Well, this is a really special place to me. I did all my graduate work out here, and it's just really a beautiful place. If you get a chance later, just kind of walk around. There's a lot to see. So before we get into our kind of heart of our agenda today, I just wanted to give folks some context for why we decided to organize this event related to mussels and Chesapeake Bay work and asking questions that we're going to ask today. I first got interested in mussels a few years ago when I went down to the Clinch River system, which is southwestern Virginia. It's a super biodiverse area for mussels and fish. And just got really interested in the work down there. Really cool stuff going on with the Nature Conservancy and DGIF doing a lot of cool restoration work to protect the species that are down there. And so this spring, I invited my colleague, Brad Krebs. I've been involved with the Virginia General Assembly's Sportsman's Caucus for a few years, and I invited him up to give a presentation to a group of legislators about mussels, to talk about what they've meant for that community. And they've meant a lot for that community. That's a part of the state that's received a lot of economic downturn, middle of coal country. And the mussel restoration project there is really just something positive that's happening in that part of the state. And there's people in that area have really embraced mussels as a mussel restoration. And that is a kind of ecotourism that surrounds it. So I invited Brad up, and the legislators, which were mainly, very rural legislators that are interested in sportsmen, fishermen, protecting wildlife, but come from, remote parts of the state, very rural parts of the state. They ate it up. They loved hearing about mussels, about things that are in their backyard. They enjoyed it so much that they planned a retreat down to the Clinch River that was a few weeks ago, and to float the river and see the mussels. First time this group has ever gone outside the General Assembly, ever met as a body outside the General Assembly. And for me, that was kind of like a light bulb moment. It really persuaded me that mussels can serve as a really valuable engagement tool for people that don't live on Chesapeake Bay, for people that don't have blue crabs and oysters in their backyard. And I know that Chesapeake Bay is not the Clinch River. Clinch River is like this super, super biodiverse place, and that doesn't necessarily exist in the Chesapeake Bay. But I don't think that was the only thing going for mussels. I think that there was a level of excitement there just to talk about something that's local, talk about an aquatic resource that's local. So that was really convincing to me, that mussels, that these freshwater mussels could play a role in getting people excited. And I think that's especially important when you think about the context of where we are in terms of the timeline of Chesapeake Bay restoration. So, this is something people have been working on for 50 years. We've gotten a lot of the low-hanging fruit, if you will. A lot of the major wastewater treatment plant upgrades have happened. And really, there's still a lot of work left to be done, but it's mainly in the non-point source sector, stormwater, agriculture, and dealing with, areas across the state that aren't close to the bay. Dealing with restoration in places like Pennsylvania and Shenandoah Valley that don't have a lot of bay shoreline. And so from my perspective, having mussels as a mascot that people can rally around related to fresh water, to connect them to local water quality, that could be a really, really valuable tool for engaging Chesapeake Bay restoration work. So that's kind of where I came into this process and why I'm really excited about mussels. In addition to all that, mussels are just really, really cool. There's just their life history and everything about them. They're just really fascinating organisms. And my background is water quality science. I was trained as a scientist, doing water chemistry, very uninteresting to most people, especially legislators. But you're sort of trained to, ignore the coolness factor about how exciting something is. But the more I've worked in kind of stakeholder groups and kind of just trying to generate support for water quality, coolness matters a lot. Having a cool story really matters. And mussels, I think, have a very cool story. And if you're not convinced of that, I hope by the time Rachel and Brian and all our other speakers are on today, that you are convinced of that, because they're certainly really great. And so, it's not just about how cool they are. They're also ecosystem services provided by mussels. In Chesapeake Bay, we for a long time have acknowledged, long before there was any credits related to oyster restoration, that oysters are good for water quality, that, they filter the water and there's some benefit associated with that. And there's all kinds of statistics about when John Smith was here, this water was filtered over and over again by huge oyster reefs. But I don't ever really hear that message with mussels. And I don't think there's necessarily a good reason for that. So, I think that there certainly is water quality benefits related to mussels. And Danielle later this afternoon is going to get into some of the science behind that and show, what are the questions remaining and what evidence is there to demonstrate that. So, last thing I just want to say before we kind of dive in is I really have 3 goals for today. So the first thing, when I think about people that work on mussel restoration, and when I think about people that work on Chesapeake Bay restoration, I don't typically think of the same person. It's almost always different people. And when I say Chesapeake Bay restoration, I mean people that their job is focused on pollution reduction. Their job is thinking about permits or MS4s or ag restoration, getting to a pollution reduction to help restore water quality, help restore the bay. I think that's a different community of people than mussel restoration people that are out there kind of stocking streams with mussels, doing research on that. So one part of today, I'm a big believer in face-to-face interaction. Is you're all here, you're all in the same room together, is to get those people talking, to get everyone talking with one another, to hear about, what is it that drives your work? What is it, the directives that your agency is under to move forward? And how might these 2 communities collaborate? I think there are a lot of ways that there could be really great synergy from the work of these 2 communities. When you think about Chesapeake Bay restoration, it brings a lot of restoration dollars to the table. Maybe not as much as we'd like, but there's a tremendous amount of livestock fencing going on around the stream, stormwater BMPs, all types of practices around the Bay watershed. And I've never really heard anyone ask the question, is there a way we could do this differently that would be protective of mussel restoration? That would be really good for mussel restoration and not influence the pollution reduction. So that's one example. I also think it could be really great at just bringing attention to the matter. Chesapeake Bay work has a big platform and there's a lot of voices behind it. And I think that could be really great for mussel restoration. On the flip side, that engagement piece I talked about earlier, having a mascot for places that don't identify with Bay stuff, I think that is what the states are wrestling with today. With the WIP process that we're going through right now, a big thing about selling Bay implementation is, well, this improves your local water quality as well. Having a mascot that sits there and says, this improves your local water quality like mussels, I think that could be really, really valuable. And then there's these ecosystem services. I don't mean to discount those. That certainly is a benefit. These are good for water quality. These are good for local water quality. So, that's kind of my first goal, is get everyone together to kind of meet one another. And then the second thing is, at the end of this, once we get through all the presentations, we're going to kind of have a chance for you all to give feedback. And I sent out 3 questions for you all to think about. The first 2 are, what concerns you about this idea? Like, are there things that make you think like, oh, that would be a disaster? Or, is there a challenge in your mind that, that couldn't happen because? I want to get those out on the table and see what they are, so we can think about them more. And then, on the flip side, what excites you about this? Is there something that, from your perspective, that, this leads to a new door that we can't really go down today? And then lastly, I want to talk about next steps. What are some strategic things that, as a collective community of Bay and mussel experts, what policies could be moved forward? I can think of a few examples. I could imagine the Chesapeake Bay Watershed Agreement, which all the executive branches of this Bay watershed states could just sign a directive related to mussel restoration. Could say that we would like to see mussel restoration occur across the watershed that we're working in. There's not necessarily any penalties associated with that, but it could be a stated goal of the Chesapeake Bay Agreement. It could be even fashioned in similar to the oyster restoration. We could have tributary goals that we hope to achieve this many tributaries for mussel restoration over the next 10 years or 15 years or if you will. Or, I could potentially imagine a situation where enough research is done on mussels to simply say, can you get a credit for it? Can you give the states credit for it? Or can you establish a BMP on that basis? I think there's a lot of research that needs to be done on that. And I'm not necessarily advocating that should be an approach that's taken. But I think it's an interesting question for discussion. And I'm hoping to kind of stimulate that discussion today. So that's my spiel. That's kind of like why I've been emailing you all over the past few months and trying to get everyone here today. And with that, I just want to start with introductions. So if you could just say who you are, what your organization is, what your title is, and then answer these two questions really quickly. How much of your work do you consider focusing on mussels? You know, is that a part of what you do? A lot, a little, it's everything I do, or I've never thought about that. And then the same question for pollution reduction or Chesapeake Bay work or eliminating pollution. You know, how much of your work is really directed at that goal? So I'll start. So I'm Joe Wood. I'm a scientist with Chesapeake Bay Foundation. I work in Virginia. I know enough about mussels to be dangerous at this point. And, you know, it's not really something I do. This is my first kind of venture into that arena. I've looked at, you know, bivalves in the past. But on the flip side, you know, bay restoration, that's what I spend all my time doing. That's, you know, like I'm 100% in that. So, Sean?
Yeah, one of the first efforts ever in the Anacostia. So we're very excited about it. I'll talk more about it.
Great. So I know that that took a little while, but I always think it's just valuable to know who else is in the room, know what perspectives are out there. And without any further ado, I'll introduce our first speaker, Brian Watson, state mussel biologist. And him and Rachel are going to kind of tag team here, both kind of getting us all on the same page about mussel propagation. What does it look like? Some basics for life history of these organisms, and what's already happening in the state. Let's talk about some of the great work they're doing already. So thank you, Brian.
I can move it, I think. Not sure where it will be best. There's probably a way to hide it that I just don't know. But
all right, I'll shuffle it around if I need to. Well, good morning, folks. As Joe said, and I said before, my name is Brian Watson. I'm the state mussel biologist with Virginia Department of Game and Fish. I've been with the agency for almost 16 years. I'm stationed out of Forest, Virginia, near Lynchburg, but I cover everything mussel-wise in the state. So all the way down into the Clinch Pile, over here into the James, and up to Potomac Basin. So I'm pretty much traveling all over the place. If you have any questions during the talk, please feel free to shout out and interrupt me. I have 15 minutes on the schedule. We've changed a little bit just to give you a heads up. I'm going to cover a little bit of the information Rachel was planning to do just on some basics on freshwater mussels. That way we weren't bouncing back and forth up here for time consideration. Worked before. Maybe stuck. There it goes. So just give you some facts about native freshwater mussels. They are a soft-bodied invertebrate with a hard shell, similar to your oysters and clams in the bay. Most live in rivers or streams. You also have some species that live in lakes and ponds. They rely on a current to supply their food. They're filter feeders, so they're mainly taking in phytoplankton, bacteria, other particulates in the water column. They have a widespread distribution. They're sedentary for most of their life. Usually once they're in a spot in the river bottom, they're going to remain there. They're not moving too much actively on their own. They can get carried long distances by high flow events, but typically they're going to be in a very small area. Have a complex life cycle. That's something Joe mentioned about mussels being relatively cool. I have some photos. I probably should have put some video up there of that, but that's usually what kind of gets folks hooked on the mussels, is showing what that life history looks like. And they're highly diverse, and they're a highly imperiled taxa. So I'll get into that. That's the main reason why we do a lot of work with freshwater mussels. So I mentioned they're highly diverse. This is a picture of mussels from the Clinch River down in Southwest Virginia, and you can just see some of the differences there on these mussels. You can see some have, some are greenish color, yellow, more of a drab brown. Some have flutations on their shell. Some have pustules. Some, like this guy here, look like a rabbit's foot. That's where it gets its name from. Some of these other mussels get their names from their shape. Monkey face, three ridge, got three ridges on it. So you can see some of the characteristics of the freshwater mussels. When you get over to this part of the states, You don't have as many distinctive features of your mussels. Usually they're all kind of the same color, similar shapes and sizes, so it's a little bit more difficult to tell them apart from each other. They live in a high variety and diverse stream systems. These are just some examples of where you may find freshwater mussels. You can be in a mountainous, rocky stream, your typical sandy Piedmont stream, your lower coastal plain black water stream here. And then you can range from these backwater swamp areas, low DO, high detritus, to James River. So you can kind of see the high range of habitats they can inhabit. Yeah, there's some species that live in streams, no wider than this podium up here. I mean, you could step across the stream. Some basics on ecology. They do have separate sexes with internal fertilization. I'll get into that here in a second. Males will release the sperm and fertilize the females. Life history, as far as how long these guys live, typically you're looking at a few decades. Some species can live up over 100 years. As I mentioned before, they are filter feeders. Primary food is phytoplankton and bacteria. They use their gills to breathe, bringing in that water constantly to get oxygen across them, similar to a fish. Habitats, again, rivers, streams, creeks, lakes, ponds, pretty much anything that's perennial, freshwater mussel can inhabit. If it's an intermittent stream, you're not going to have mussels there. Now, you can have a perennial stream that goes dry in the summer and have mussels still be in that stream. We've seen that in numerous streams go dry in the summer. Mussels will burrow down as long as they're in a wet area. They almost go into a hibernation state and they wait for that water to come back, and then they come back to the surface. As far as pollution tolerance, depending on the species, they're considered intolerant to moderate. So this is an example of the life history of freshwater mussels. Most freshwater mussels require a fish host to complete their life cycle. So what they have, you'll have your male and female. This is your adults here living in the stream bottom. Sometime during the year, males will release sperm. Females will siphon that sperm in, and they'll fertilize eggs within the female. And those eggs will develop into a larvae that we call glochidia. That's this guy here. I'll show you some photos of that. They're usually about sand grain size. I tell folks they look like a little Pac-Man. They kind of sit open, and they're brooded in the gills of the female, and they have to get those onto fish. Some species are generalists. They can get them onto any number of fish species. Some species are specialists. They can only get them on maybe one species of fish or a few different species of fish. So if they happen to get on that suitable fish host, they will be on the gills, maybe the body, the fins, and they will stay on that fish anywhere from a few weeks. Sometimes they'll overwinter on the fish, and they'll transform into a juvenile. They'll develop some internal structures that have that foot in there. They'll have gill development, and they will drop off. And if they are in the right habitats, they'll grow and become an adult. So that's kind of the general life cycle of the freshwater mussel. There are some species that can skip this stage. They have direct transformation. So those larvae will develop directly in the juveniles within the female. And there's any number of ways that these mussels try to get that larvae onto their host. This is just some photographs. We consider some active or passive, where the mussels are actually trying to attract the fish to come to them. And these are just some examples here. Here's your mussel. This is what we call a superconglutinate packet. That is all those larvae packaged up into this kind of lure that the mussel spits out and holds onto with the mucus thread and kind of fishes out in the water column to try to attract a sunfish or a bass to come and eat that. Instead of getting a prey item, it gets a mouthful of parasites. This is another mussel here, acts like a Venus flytrap. Has little lure inside of the shell there. The fish sticks its head inside there, and the mussel grabs the fish, holds onto it, and pumps the larvae onto the fish. This is a more common one you may see videos of. The inside of the mussel, they have the mantle tissue on the inside of the shell. Some have that modified into a lure. This is that lure here. You can see the eye spot. Almost looks like a fish. They'll wave that back and forth. And right up here, you can see those little structures. That's the gill. So if a bass comes in and strikes at that, the mussel will release those larvae. And then this one here is the whole mussel sitting up on the surface. and it rocks back and forth and looks like a crayfish to try to get a fish to come to it. Another conglutinate packet release. This is more of a passive infestation process, a kidney shell. Here's the gills. They have this little conglutinate, looks like a black fly larvae. They release them out in the water. They have a sticky pad. They sit on rocks in various areas of the stream. A darter may come along like you see here. I think it's got a black fly larvae. Eats that guy, it ruptures, and they get all the glochidia go across onto the gills of that fish. So here's some photos of some glochidia. Said they're usually anywhere in size from about 50 microns to about 300 microns in size. So size of a grain of sand or a grain of salt. You look at them in a petri dish and you just see little specks there. But this is what they may look like, little teardrop shape. Here's one here from a particular species. You can see it's got a little tooth on the end of it. These guys can hang on to the body or the fins of the fish better than the ones without that hook-like structure. And then here's an example of them actually on the fish gills. And they will actually insist into the gill. That fish has an immune response to those glochidia. So they will reject some even if they're suitable hosts. The ones that stay on insist into the tissue. As I said, remain there. It's water temperature dependent for some species, may drop off in 2 to 3 weeks, may overwinter. And then there's your juveniles. Kind of look very similar to the glochidia. There's not much difference in size or in the shape of those guys once they first come off the fish. And then there's the foot sticking out from the juvenile mussel. Usually during about the first month or so, they're pedal feeding at that time. The gills aren't quite fully developed in these juveniles, and so they kind of creep around picking up sediment and different things initially to get bacteria and particulates off to feed off. So why are mussels important? This is usually a big key component. They're great indicators of water quality. Anytime you have an organism that is sessile, doesn't move, and is constantly in contact with that water, great indicator of water quality. They're long-lived, and some of these species are fairly sensitive in early life stages. As David mentioned, new water quality standards for ammonia, those all came about because of toxicity test work with freshwater mussels. The organisms that were being used for ammonia were not nearly as sensitive as freshwater mussels were. So through this process of doing the propagation and being able to get juvenile mussels, those were added to the tox work, and so that's helped drive some of these new water quality standards. Filtering capacity of these freshwater mussels. This is a great demonstration we usually show. We have 2 tanks, one no mussels, one with 10 freshwater mussels. It's about a 10-gallon aquarium, and this is over the course of about 90 minutes. So you can see that tank on the right starting to become more clear. And then after about 90 minutes, it's almost crystal clear, where you still have particulates up in the water column on this left-hand tank. So you're looking at certain species can filter up to half a gallon to a gallon of water an hour, depending on how active they are in their size. So tremendous filtering capacity. So when you hear historically about how the oysters used to be turn over the entire water volume in the bay in a certain period of time, freshwater mussels were providing the same function in our freshwater streams and rivers. And Danielle will get more into that in her talk, I'm sure. These guys are important for wildlife food sources. You have things like otters and muskrats and raccoons feed on these quite regularly. Certain turtles, certain fish like some of these suckers here. And then also you got the non-native blue catfish. That's a small part of its diet as well. They're also great for habitat for various other aquatic animals and plants. You can see here there's certain mad toms that will use the shell material to lay their eggs in. They're pretty specific as to the type of habitat they want to lay their eggs on, and they'll target some of these mussels. A lot of times you pick up the mussels, you see aquatic insects on them. It's not uncommon to see a higher diversity and abundance of aquatic insects in an area where you have a healthy mussel bed. Same with the fish diversity and abundance. They all kind of go hand in hand. Historically, freshwater mussels were used for button production. This is just some old photographs of the Muscatine Button Company. they would harvest the mussels and use the shell to punch the buttons out of before the development of plastics. They're still used today for culture of pearl industry. There's some states that have harvest of the mussels, and so what they do is they collect the mussels. They will punch these beads out of the mussel shell, insert them in the oyster, and then you get the cultured or freshwater pearls. And so it's not as prevalent as it once was, but there is still an industry for that in places like Tennessee and Kentucky. And those are usually shipped over to China where they do this work. As far as diversity of freshwater mussels, there's about 900 species worldwide. And if you look at the distribution, North America is the hotspot. We have about 300 species. This number's up a little bit. The slide's a little bit old, but there's always changes in the taxonomy within this group. So we're about 300, a little bit over 300 now. But you can just see, looking at some of the major areas, how much more diversity we have here in North America than some of the other continents. And if you look in the United States, not unlike any aquatic diversity map, the hot spot is in the southeast. You'll see that with amphibians, reptiles, fishes, almost any aquatic organism. Virginia, we have about 81 species depending on taxonomy, and that ranks us about 6th nationally for the diversity. So within the states, the hot spot, you've heard some folks mention the Clinch, the Powell, Southwest Virginia. That would be down here in the upper Tennessee basin. It's blocked out, but there's 31 endangered and 3 threatened species down in that watershed. So that's why there's been a lot of work focused on Southwest Virginia for mussel restoration over the past 2 to 3 decades compared to what we've done in the Atlantic slope. In the United States as a whole, the conservation status, you look at the number of extinct, endangered, threatened, and special concern species, about 3/4 of freshwater mussels are at some level of impairment. So only about 1/4 of those species are considered currently stable. And that's mirrored in Virginia. So this is the number of federal T&E, SGCN is species of greatest conservation need. Then you have state endangered, state threatened, and you're stable. So you're about the same percentage, about 25% are considered stable within Virginia. So you see your mirroring of what's going on at the national level. And if you compare those to some of the other groups, how do mussels rank as far as impairment status? They're pretty much up at the top. If I were to throw aquatic snails on there, they would be right up there with the freshwater mussels. But you can see it compared to things like birds and mammals, butterflies, things that get a lot more conservation dollars, that are considered more charismatic than freshwater mussels, much more highly imperiled than these groups down here. And so that's the message we're trying to get across. Most folks don't realize some of these mussels even exist out in the rivers. And so if I were to put up a chart here to show where the dollars were going for conservation, it would be flipped on its head. You would see the things that have the least level of impairment getting the most dollars typically. To give you an idea of mussel propagation in Virginia, kind of when it started and how we got to where we were starting the facility down the road. Mussel work and propagation has a long history. You go back and look at some of the old papers in the early 1900s. There was a good amount of work going on, and then there was a few decade lag there where there wasn't a lot of work going on in the mid-1900s. But in Virginia, mussel work kind of started up in the 80s. Mine, Catherine's major advisor, Dick Neves at Virginia Tech, kind of started the mussel work here in Virginia, and that kind of went on through the mid-90s. There was a lot of different work he was doing there with graduate students coming in at Virginia Tech. And it was about in the mid-90s when he started the propagation work. I started there in 95. And the facility they started up there kind of started in 95. There was really not much propagation work going on before then. It was all life history work and different aspects regarding freshwater mussels. A lot of those projects were mainly funded through Department of Game and Fish and U.S. Fish and Wildlife Service dollars. And then in '96, they started, which didn't have the name at that time, but the FMCC, the Freshwater Miles Conservation Center at Virginia Tech. That's when they kind of kicked off doing actual propagation. And this is an aerial photograph of what the facility looks like now. At the time, they had this little space in the greenhouse at one end of it where some of the students were doing work. This is the fish aquaculture building here. And we had some space at the end of this building. That was what they had for the students doing the propagation at the time. Now they've kind of got this whole facility down here that they've grown into over the past decade with the pond that provides their water. And they've actually have most of the fish aquaculture building where they do mussel work in now. So since '96, where they had these little spaces at the end of these buildings, they've almost taken over this whole facility here. Also in '96, my agency hired a full-time state malacologist, to focus on freshwater mussel work. And then in 1998, we started the Aquatic Wildlife Conservation Center, which is in Southwest Virginia. That's down in Marion, Virginia here. This is a picture of the facility. Basically, it was old fish raceways that had some structures over top that we converted into labs and buildings where we could do mussel work. If you've ever been to a facility, a lot of times it's not high tech, it's not fancy. It's usually trying to pirate in a piece of property that is no longer being used, and that's what happened here. And to kind of show you what it looked like initially, that was in the early days, so you can kind of see it's pretty defunct. And still today, we're trying to move into a new building we've been trying to do for about a decade. You got leaky roofs, and you're almost like working in a submarine when you're in the lab over here. The ceiling's about 6 feet high, so you're kind of crouched down in some areas. And then in 2007, get that out of the way, that's when we started the facility you'll see later, the one Rachel runs. That's Virginia Fisheries and Aquatic Wildlife Center. That was a partnership between Department of Game and Fish and the U.S. Fish and Wildlife Service to start a freshwater mussel propagation facility that focused on the Atlantic slope. The work being done at Virginia Tech and the work being done at AWCC was all done primarily in the upper Tennessee Basin, because that's where most of the endangered species were. You're endemic, so they were focusing down there. But over the years, there became more of an interest to look at Atlantic slope species as well. I think I may have froze it, Joe. Come on. There it is. So this is just an aerial of our facility down the road, which you guys, those of you taking a tour, will see later. And we were kind of down on the end of the facility here using the ponds. Rachel will get more into the details of what we do at the facility. But to give you an idea, didn't mention with AWCC, since 2003, they've produced about 6.1 million freshwater mussels, and they've released, I think it was about 70,000 individuals that were taggable size. We talk about taggable size, you're looking at about 2 centimeters, about an inch. Since 2007, we've produced about 8.6 million mussels and released about 200,000. So why the Atlantic slope? As I mentioned earlier, there's 41 mussel species listed in Virginia, 34 in the upper Tennessee, only 6 in the Atlantic slope. However, if you look at our wildlife action plan, which looks at those species of greatest conservation need, about a third of those mussel species fall in the Atlantic slope. So that was the reason for focusing and trying to develop a facility that was looking at the Atlantic slope species. And outside of what we've been doing here in Virginia, not many other states were doing any work on the Atlantic slope either. North Carolina was probably the only other state doing any Atlantic slope species work before we came on line here. And I know Rachel was doing some when she was at White Sulphur Springs. They were doing Ohio Basin River stuff as well, and a little bit of Atlantic slope work, but there wasn't a real focus on the Atlantic slope probably until about the past, I would say, 8-9 years. And this is kind of the goal of what we're looking to try to get out there is larger mussels, cooler fulls of mussels, trying to get those out in the river. This is one of our early releases we did on the Nottoway River down in Southampton County. Early on in mussel propagation, a lot of these individuals were released when they were small. They were less than a millimeter because you couldn't grow them past a few weeks. But over the past 10 years, we've developed the techniques and technology to be able to have grow these almost indefinitely at our facilities and get them up to a large size. And we see a much better return when they're larger than when we're releasing them after just a few weeks. Just to give you an idea of where some of the funding comes from or did come from for this type of work. Initially, a lot of this was Section 6 money, which is a pot of money that focuses on federally endangered and threatened species. Also, state wildlife grants monies that looks at T&E species and those species of greatest conservation need. Various mitigation sources. Some dollars we got through VIPD's permit renewal from Dominion for their Bremo power station. That provided a lot of funding for starting Harrison Lake Hatchery down the road. And unfortunately, a lot of times we rely on these natural resource damage and restoration settlements. So it's like chasing the calamities to try to get dollars. We don't have a set pot of money that we tap from. So what funded AWCC and some of the Virginia Tech work was these Lone Mountain and Sturtis. This was a coal slurry spill in '96. The dollars came in '01. And Sturtis was a tanker truck spill in the Clinch River in '98. The dollars came in 2004, and this was the amount of the settlement here and the amount of money that went towards the mussel restoration components of that restoration project. And then in 2017, there was a settlement with DuPont for South River mercury damage up near Waynesboro. $42 million, and $4 million of that is going to mussel restoration. That's a significant chunk of what's providing some of the dollars for us now at Harrison Lake. And then they have the coal, I didn't mention the coal slurry spill in the Dan River in 2014. It's things like that that we unfortunately have to rely on for the work that we do. And that's it.
So any questions? Brian, before we move to the next presenter?
That would be anything, if a drop of rain hits the ground and it falls towards the Atlantic slopes, that'd be basically your the fall line. So if you are east of the New River Basin, if you're in the Roanoke, the James, the Shenandoah, many of those watersheds, you would be in the Atlantic slope basin. The New River goes to the Ohio, and then everything west of that's into the upper Tennessee. Mm-hmm. Both. When we first started, it was targeting areas that had mussel populations, and we were focusing on the species of greatest conservation need to bolster their numbers. But now with this South River DuPont settlement, there's areas there where mussels are gone from because of the mercury contamination, and we're potentially looking at targeting some of those areas to see if we can restore mussels. The habitat looks good. Mercury's probably still there. We know it's still there at some levels. So we're looking at maybe working with the common species to get back in there and see if we can restore some of those areas. But usually there's some mussels at the site that we're looking at for restoring.
Outside of the mitigation efforts, what is we think the need is for funding? Both staff and already all of the river and stream.
How much? Yeah. I don't even know if I could put a dollar figure on that. Any ideas, Rachel? That might be a question for later.
I'm gonna talk a little bit about that. But I would give you some time.
Yeah. Because I could tell you for what we have at Harrison Lake now compared to what we started in '07 is a pretty functional, high-staffed facility, and we can't touch what needs to be touched in Virginia and the Atlantic slope. We can work in areas, but we just can't. There's just no way to cover everything. And I'm not sure if there's a funding amount there that would be available to do that.
So sort of a different way of looking at that. Am I right that there's really not hardly any stable funding that comes to this program aside from mitigation dollars? Maybe staff time, but
yeah,
the state invest.
The state does. We invest the state wildlife grant dollars that come in that mainly funds the non-game work through Department of Game and Fish, but a lot of it is generated off of mitigation dollars. There's not a stable source that we can tap into. And the state wildlife grant dollars, a lot of that are already eaten up by staff time and other things, so there's not a significant chunk left there in our budget to say we're going to use X dollars for mussel propagation work and devote it there.
Service does get money, some money for some of the equipment and maintenance infrastructure. Because my salary is my own salary project. Some, but it doesn't nearly cover. You have to cover and find funds for the rest of it.
Which the facility produced something like 8 million individuals for release was second that just reflect the mortality rate.
Yeah, and Rachel will get more into this, but a lot of that when you first start, it takes a few years to get a good idea of how to culture these mussels to a size. So you'll see a significant amount of mortality. Sometimes you might have 100% the first couple of years that you're working with the mussel species, and typically it's going to be high anyway. Even if you're doing well, you might get 20-30% survival to a size. Now, initially, as I mentioned, when this work was being done, folks were releasing large numbers of mussels that were a few weeks old, but they were seeing little to no return on those mussels. Now we do see a return when we're growing these mussels for a year to 2 years. Being able to tag them, we can go back out of seeing mussels that have been released 6, 7, 8 years later. We can track their size. We're seeing them spawn in the wild as well. Almost every species we've released from the hatchery, we've seen spawn in the wild, which is kind of one of our end goals for success determination when we're looking at these types of projects, aside from moving the needle with the numbers out in the wild. Usually they're in their second year of growth. Yeah, sometimes we can release them at the end of the first season if they're dependent on the conditions at the hatchery. And if we get them off early enough, usually they're going out that second year. Yeah, varies from site to site, and sometimes if your site's not as stable as you would like, trying to recover those is difficult, so you don't know exactly what happened to those. Some sites folks have seen where and Rachel will get into this on tagging, you might see 90% recovery over time. That's some of the things we're trying to look at, but the larger rivers like we work in, like the lower Nottoway, because of the size and it's black water and difficult to sample down there, it's hard to get a great handle on. We might release 20,000 individuals over a long area, trying to recover those. We've got to kind of estimate what that recovery is, but if your site's stable, you should be getting 60, 70%, at least I would say, of those animals surviving that aren't going to die naturally.
So, that round of applause for Brian. Thanks. Now over to our next mussel expert. Rachel's going to talk about some of the work at the hatchery the U.S. Fish and Wildlife does. Thank you very much.
To move forward, so we have a laser pointer or something.
Yeah, I'm having some issues with that. But yeah, so it should be left to right, but you can just use the mouse for the mark. And if you need to move this, you just kind of drag and drop.
Great. Okay. So I'm going to go into a little more detail, left off where Brian kind of left off, and talk about specifically methods, techniques, some of the work that we do at Virginia Fish and Aquatic Wildlife Center at Harrison Lake National Fish Hatchery. So a lot of people have mentioned the Clinch River. That's where I got my start 20 years ago. If you don't think mussels are cool after today, then I encourage you to get a snorkel, stick your head in the Clinch River in Tennessee, And if you don't think that's amazing, then I don't know, there's no hope for you. So, where do I point?
Actually, that's
Oh, okay.
You might have to sit on the screen with a mouse to do that.
Okay. I'll get it by the end. So this is just kind of a snapshot of current mussel propagation facilities in the US. The red are facilities that have been going on, that have been producing mussels for over 10 years. There's a lot of up and coming. Maryland is starting to do some propagation. Seems like every day I hear of other facilities coming online, usually because of a big settlement that brings large funding stream their way. Pennsylvania is talking about getting another facility. So there's a lot of interest right now, a lot of funding trying to be used for freshwater mussel propagation. And then you can see Virginia is really pretty big. It's got more propagation facilities than any other state. Oops. Can I go back somehow? Here we go. Okay. Just wanted to make sure I didn't miss one. So this is kind of where we are. We're literally a mile and a half away. We're in this wonderful little part of Virginia. And you can see this is Harrison Lake. And our water, we get our water for our ponds, which we culture a lot of mussels in our ponds at the hatchery. That water is gravity fed from the lake to our propagation ponds, which have been used for a wide variety of fish in the past, but right now we use 4 for freshwater mussel production. Our lab, Brian showed you a picture of our lab, and everything is right in here. Brian also talked about the life cycle of the freshwater mussel. That can make it challenging when you are trying to produce juveniles for these animals. So you need a host fish to complete their life cycle. To propagate them, we go out and we collect females that already have the larvae in their gills, so their eggs have been fertilized. We bring them back to the hatchery, and we either extract using a hypodermic needle and a syringe filled with water and flush the gills that have the larvae in them. We either do that, or we let the mussels just release on their own, release conglutinins or release larvae on their own. A few species will do that we work with, but typically their gills are made to be ruptured by fish. We've tagged animals that we've extracted with a needle and a syringe, and doesn't seem to affect their survival. Their gills regenerate. So that's usually the method that we do. So once we get our larvae, we put them in with fish anywhere from 5 minutes to an hour. The larvae will attach. They'll be on the fish, on the body, the fins, the gills, the eyeballs, and they can attach. Some species can attach anywhere. After 2 weeks to a few months, the mussels will drop off and exist from the fish. And then we have various methods that we use to grow them out and culture them. So the first thing that we do is we go out, we collect our brood stock. There are 2 types of brooding females. There are long-term brooders, which means that they become gravid in the fall of the year, and they hold their larvae in their gills all the way till the following spring, where they release and try to attract host fish naturally. Short-term brooders are much more difficult to work with. They have a short brooding cycle, so they will become gravid late spring, and they'll release their larvae naturally in the wild in early summer, midsummer. So to do this, we sterilize. We're big on sterilizing all of our gear, making sure we're not transporting things from stream to stream. We use aerated coolers with or without substrate, maintain temperature, because mussels can abort or release their larvae if the temperature increases or decreases when you're traveling back to the facility. So keeping temperature maintained is important. And then we check for gravidity by looking at the gills of the female. So this is a non-gravid gill. It's flat. And then you can see a gravid gill is really puffy that has the larvae in them. Some mussels, only 2 of the outer gills will become full of larvae. Other species, all 4 gills will have larvae in them. So we extract, we use a hypodermic needle and a syringe to insert into the gill. So we'll insert the needle, will come in all the way through the gill and then flush with water, and all the larvae will get flushed out of the gills. Other animals release conglutants, which are these little white these are packets that contain larvae. This is the James spiny mussel. It's federally endangered. So we're letting it release naturally on its own. It's releasing these conglutants. The conglutants, closer up, they look like this. You can see the little dots. Those are actually the larvae on the outside of the conglutant. The conglutant mimics a worm or an aquatic insect that attracts the host fish. In this case will be chubs, mountain red bellies, rosy-side dace, some basic your minnows. And then under the microscope, a little more magnification, you can see the larvae for the James spiny mussel. So they're laying open. They're kind of protected in this little shell or membrane, I guess. When they come in contact with the fish, they snap shut, and that's how they attach. So to infest, we put the larvae in with the fish. These bass, usually it'll be 5-10 minutes. Heavily aerate it. We check the gills periodically to make sure that we're not putting too many larvae on the fish, because that can cause some mortality. And then we're looking for the end result where we have a lot of mussels on the gills of the fish. After the infestation, we separate fish by species or size. We place them in an aquarium. They will drop off 2 to 4 weeks at 20 degrees Celsius. We have sieves that are on the tanks that the juveniles will wash into. We'll collect those, or we'll siphon the tanks, count, record, and we'll put them in just various culture systems, which I'll talk about. There's a lot of techniques, options, and involvement. You can do stream-side or free releases, which are putting larvae on. You go out to a site, you collect fish, you collect mussels at that same site, you extract right there in the field, you put the larvae on the fish, and you let the fish go. You can let the fish go in another area, the same area. You can't tell if you've done anything over the years unless that animal is extirpated from your site. So there's no real idea of success when you do those. So what we usually do is we like to tag individuals that are larger in size that we've grown out for over a year and release those larger animals. We teach a class, myself and some colleagues, at the National Conservation Training Center at NCTC. There's a propagation class in September. If you're interested in learning more about how to actually do this, I would suggest taking that class or come and talk to me, do another tour. So juveniles, when they drop off the fish, they're very vulnerable. Predation, we know that they're vulnerable to being handled or handling stress. They can float if they want to. They can puff up their mantle tissue with air and they can float. So you need to watch for escapement. Overfeeding, they're very sensitive to ammonia, like we talked about earlier. They're sensitive to having too much food in the water. It will cause them a lot of stress to have a lot of food. They can't process it all. So that can be stressful to the animal. Water quality, and of course, poor nutrition. So we've come a long way. Somebody asked the question, I was running to the bathroom real quick, but back in 2005, even getting a picture of this was rare. I mean, this was woohoo, we got it, 2005. I can see the mussels that we're going to release, because before then it was primarily microscopic, you know, and now it's just every year we get better and better. 2014, 2016, 2017, you know, some species were really able to move the needle. So when we talk about survival over time, and we're just now getting to the point where we can produce, for some species, we can produce a lot of mussels and we can grow them. These individuals were grown in 4 months for this one species that's a fast grower. So we're finally getting to the point where we're really able to pick our restoration sites, plan numbers, and see how many animals we want to put back in there, and go tag them and go back and monitor and find them later. We're finally getting to this point, which has been 20 plus years in the making. I have too many things I'm doing here. So how do we culture juveniles? During the first 2 months, when they're very small, there's a lot of mortality. We're getting better at this every year. These animals are mostly pretty high fecundity, so Mother Nature has pretty much planned for there to be high mortality when they first drop off of the fish. Once they get a little larger, at about 4 months, mortality kind of goes away, especially when they're a millimeter or over. Mortality is slowed down significantly at that point. The systems that we use at my facility, we use buckets, rearing pans, static aquaria, and I'm just going to briefly kind of go through what we use to grow mussels. So the buckets are developed by Chris Barnhart in Missouri State. It's a downweller, basically. We use upwellers and downwellers in freshwater mussels, as you guys do for marine bivalves. It's a very small footprint. It's recirculating 2 5-gallon buckets right here. So water is being recirculated to through these 2 buckets. There are chambers which have nylon screen in them. When the mussels start out in these, the screen is 150 microns, and as they get bigger, you have to change the screen, so that gives them more food, more flow. The screens get clogged, so there's a lot of maintenance. They're pretty labor intensive, these systems. You have to change the water, spray them down every day, clean the screens, change the water every week. And then as they get bigger, put larger screens on them. But they are great for research. We do diet studies, we do temperature studies, we do a lot of research at our facility as well. So this system, although labor intensive, is useful for conducting diet type studies. The rearing pans, once mussels get a little larger, we use these pans that were developed and started being used at AWCC that Brian talked about earlier. It's flow through pond water. So they've gotten past their really sensitive time, and we're able to give them flow through pond water, which has predators in there. So we have to wait till they get a little larger to put them in there. Once we get them in this type of system with that pond water, it's got a lot of good food in it, they start to grow pretty quickly. It's great. It's low maintenance. We don't have to change screens. We don't have to do water changers. We're not relying on a bunch of little pumps to keep them going. So these systems have worked out really well for us, and we'll show you these later. And they are happy as a clam. Filter, filter. So the other system we use, it's just a static tank. You can use a 5-gallon bucket. You can use a 10-gallon aquarium. You really can use anything that you can put an air stone in, and a thin layer of fine sediment, less labor intensive. Again, small footprint, easy replication for studies that we like to do. You don't really need any specialized equipment. It's short term. You can't leave them in the system like this for very long, 2 months. And then we usually move them outdoor to get some of that magic pond water. So anything involving ponds and using pond water, we get really good growth and survival. They're low maintenance, lots of different options out there. The mussel world has expanded its culture capacity and the systems that we use pretty exponentially. We use floating baskets pretty regularly at Harrison Lake, which you'll see in a little bit, they have a lot of benefits. You don't have to maintain them. Figure out what I need to do here. You don't rely on, you don't have to grow algae. You don't need to buy algae. Mussels are needing more than just algae too. They get bacteria, they get all kinds of yummy stuff from the pond water. Growth rates are significantly higher. We can put tags on some animals within 4 months. They're large enough to put a tag on. Again, it's less labor intensive than indoor culture, but you do need ponds. Advantages of indoor culture would be we're keeping species separate, we're keeping river drainages more separate. We can control the culture environment when they're outdoors. It's, you can get predation, you can get all kinds of things happening outside. So, baskets were developed at Harrison Lake. Great growth and survival, low maintenance, low effort, low manpower, low cost. Win-win-win. We can get them out of the hatchery, and that's a good thing. So it's a fish spawning basket. Maybe you've seen these orange baskets. We put a layer of nylon mesh at the bottom, put an inch or two of sand, rinsed sand, and then that's kind of what they look like, the finished product. Lots of pool noodles. We have a lot of pool noodles. It's a beautiful, colorful pool noodle pond. So we also put a top on it and lots of cable ties. We do have muskrats. We have animals that want to get in there, so we have to keep them safe. So we use anywhere from 3 to 10 millimeters. We'll put mussels in the baskets. Some species we can get 200-300 mussels in a few days that drop off of the fish, and then we'll start, depending on space, we'll start moving them into baskets earlier. We put them out when the water temperatures begin to warm in April and May, and they stay in the baskets till the following year when it starts to warm up, and then we'll remove them, put a tag on them, and release them into a restoration area. So this is kind of what our ponds look like. When we grow mussels inside, mussels are eating constantly 24/7. So when we're growing them inside, we have automatic feeders we like to use, timers and solenoid valves. The length of time that the solenoid is open and distributing algae is controls how much algae is being distributed. We feed indoors, we feed a diverse diet. We use several microalgae, shellfish, Nanochloropsis, and phytophthis are purchased from reed mariculture. We also use a lot of our pond water, which gives them a lot more of a diverse diet than what we can purchase. And then we monitor food using a Beckman culture counter. We look at food quantity and quality on a regular basis. So a lot of work goes into this. This is a tremendous amount of work. Indoor systems every day, we inspect the mussels, we clean the systems, we spray the screens, we look at them, we look and see if there's food present in the gut. You can always tell if they're happy because you get this, it looks like a black dot, but that's green, that's food in their gut. And so that's something that we always look for every day. We want to look for any shell material in the systems, make sure they're not dying. Look at growth, are they growing? You can see here, we're always measuring our lengths on our juveniles. Make sure everybody's healthy. Outdoors, there's not a whole lot we can do. We try to stick our hands in the baskets as often as we can. There's points in the summer where we have 200-300 baskets out. So just checking on them when we can, looking at growth, looking at how good they look, shell material, things of that nature. Once we have grown them to a taggable size, we use several methods to tag them before they're released. Black super glue is something I don't have a picture of, but it's this rubberized super glue. We just put a dot on the mussel, and then we can find them later in the river. And if they have the dot on them, then we know that they came from us. Usually we use glue-on shellfish tags or hull print tags. So we record the length of the mussel when it's released, the number, the tag number, the color, and then when we find them, we can tell how much they've grown, and we can get a little more information off of each mussel that was released. We also laser engrave our mussels, which is awesome. 2 people can laser engrave a couple thousand mussels in a day, which before the laser engraver, that was one of our biggest staffing needs, was tagging. When you have 50,000 mussels to tag, that's a tremendous amount of time. So the laser engraver has helped us a little bit. We laser engrave the year, specific number, letter, number combination. We measure the mussel, we record all of this data for later. And then we put, because we're concerned about erosion to the shell, when you're laser engraving, you're removing the very top layer of periostracum or shell material. And so we coat that with basically nail polish, clear nail polish or nail glue. And that protects the shell from more erosion. And pit tags, when we have a rare species or an uncommon species, or ones that we really want to increase our ability to find them later, we use we use pit tags, which we super glue onto the shell and then cover that with dental cement. That is the white that you see, and then beneath that is the pit tag that you can't see. But we can go out and use a pit tag reader in the stream, walk along and hear the beep, and then try to find the mussel, pull it up, get the data, get the tag number, see how much it's grown, things like that. So current projects that we're working on, Brian talked about a few of them. We have a NIFWIF funded project from a coal ash spill in the Dan River. We're working in Virginia and North Carolina on this project. That project is 4 years. It'll end in 2020. And that involved propagation of 4 species. It involves moving adults into the restoration area. So we're moving Elliptio campionata. We're pit tagging them, about 250 adults a year. We're testing our site suitability for releases of juvenile mussels that will be occurring in the next year or so. We're doing some genetics projects. We're looking at broodstock from different places, if there's any genetic differences in those that species, so we can release it into the Dan if there's no differences. We're also looking at a genetic effect of hatchery reared animals versus the wild population to make sure that we're not skewing the genetic material in those animals that get released. We're also looking at some diet studies using DNA to see what these smaller animals are actually consuming. And we're doing that through Lamar Fish Tech Center in Pennsylvania. It's a Fish and Wildlife Service laboratory. Here's our campanata pit tags and our first release we did last year. We have a bunch of small projects. Dominion Power, not really a small project, but we're doing propagation of 5 species, genetic study for implicata. We have a new project with the Forest Service that we're working on, James spiny mussel to augment in the cow pasture river. That's the James spiny mussel, some juveniles right here. And then we have a large 10-year project for the South River, which is part of a DuPont NERDA settlement. And that will be a 10-year project. We got started this year, and that involves propagation of 7 species. And we have an in vitro lab that we've set up here at VCU Rice River Center. They were nice enough to basically let us use a big lab here, which is great. If anybody's interested in seeing that setup, we can walk over there. It's two doors down. So there's some species that we don't know the host fish. The host fish is hard to find. So in vitro mussel propagation has been coming online for quite a few facilities in the past couple of years. So we started our first trial last week
And we plan to do that for 4 species for the South River project. Let's see. So last year was a fairly typical year. We produced a little over 1 million juveniles. The year before that, anywhere from 1 million to 2.5 million has kind of been our total numbers. So you can see we work on anywhere from 10 to 12 species. We work on a variety of rivers, anywhere from Atlantic Slope, James River drainage, Dan River drainage, Nottoway, the Chewan River system. And you can see survival. I mean, this is for mussel culture, this survival is good. And you can see it varies with some species. But as we start doing more and more species, it takes a few years for a new species to start learning its suitable culture conditions till we can get to a point where we see increases in survival. It takes time.
You have 10% survival for winter, but then you're saying once you get them that far out into the ponds, then you've got that.
Right, and it's hard to tell because they're in the ponds for a year. So we have to wait, these animals, we have to wait till this year when we start tagging everything from the pond to get our survival. That's why it's not complete. So we have survival to winter, we have indoor culture, and then as we go through the following year and start pulling our mussels out, we can fill in some of this data. Yeah, if you have questions, feel free to. So last year we released 45,000 mussels. These are mussels in the 20 to 40 millimeter range. They're large. They have been laser engraved, pit tagged, or hull print tagged. And so here you see some basket survival as well. So it's averaging about 40%. And some of these are like implicata, the top one here, and these are released usually when they're very, very small. So I started looking at, okay, we can produce hundreds of thousands of implicata. They use blueback herring as their host. And so we can literally produce hundreds of thousands a day when they're dropping off. When you have that many numbers, you can't possibly keep them all inside. So you have to get them out. So we're putting in the baskets when they're a day old. We're not growing them at all inside. And so the survival is still, I mean, that's pretty good for a day old juvenile in a basket in the pond. Yes?
Are you keeping a population that you release out to the river and pond during birth rates, retained in the river ponds and stream population?
We've talked about that. There probably are some because we have so many baskets, we can't get them all out. But I don't know that we've analyzed that data yet. Yeah, of course.
It's debating with kind of common cohorts by size as opposed to academic sacrifice tables.
What do you?
So I guess if you had this early survivorship goes up after the first winter and then you release them into the stream, you have the recovered specimens. Are you able to follow the ages well by size when you actually do put them out in the stream, whether that small amount you recover? Do you think that it's something that you can put a wild population alongside and kind of compare, kind of like working on the trees?
Yeah.
Is there any potential for that? I know that's kind of a long shot because of variation in the habitat, but
I'm not sure what you're asking.
Okay.
That survival of the wild compared to the stock.
well,
Same age,
yeah,
class,
survival,
class.
more so than survival is trying to figure out some of the species that exist for a long time in a particular area. Looking at trying to use the size and potential, whatever morphological characteristics might be there. Looking like drought years, flood years, things like that. And since you have this, the pond that you can kind of control for, how they're growing artificially, it's at least kind of a benchmark. And then when you release it into the stream, warm year or cold year, it's the pond seems to mature. It's just, yeah, trying to almost look at just like a tree ring.
Yeah.
If you've had any luck with field analysis.
I don't know if you know of anything that's been done.
No, yeah, I don't. Because it's when they get to a certain size and become reproductively mature, you don't see a lot of growth at that point.
Okay.
You'll see a lot of growth initially the first few years, especially with our mussels. We can have them to reproductive size sometimes at the end of that second year we see them start responding. So when they get to that size, even in the wild, coming out of the collection, some mussels will pass recast them 14 years later, they're around like 2 millimeters, 3 millimeters. So they're just, everything's almost going to reproduction and kind of sustaining that size that they're already at. So it's hard to tell at that stage anything growth.
Okay.
We do see, you know, when you look at mussels, you can tell approximately how old they are by looking at the shell growth.
Yeah.
And so we do see sometimes where we've had a really cold year or something like that, we do see the growth ring. You can look at the mussel visibly and sense that's been happening. Compared to the wild, I mean, the ponds are pretty similar in temperature as the wild, if not maybe colder at times. Catherine, did you want to add?
Yeah, I just wanted to add, there have been some studies out there, master species and PhD species, that looked at shell growth and compared it with, you know, the history of what's happening in the stream. So there have been a lot of studies that are analyzing shell material and comparing it to whatever chemistries in the water, looking at what's happening in the groundwater. So we do know that, you know, your growth is affected, just like what Rich was saying.
If you take a cohort and put it into a pond as a reference, it would be a good growth of pond, it's more nutrition. You put it into a variety of candidate restoration schemes, same cohort. And then you measure growth rates over a period of time comparatively to your reference. In the streams, there's rarely as much food-rich conditions. And so the growth will always be low.
Yeah.
And you can get a barometer of the relative viability of the different streams for getting good mussel growth. But as Brian said, once you get the reproductive age, they're going to want to start switching from somatic to reproductive. You know, sometimes if it's a stressful stream, they might want to put all their energy into reproduction as opposed to growth. So you start getting wonkiness once they get reproductive. But early on, growth rates are a really useful measure of the relative suitability of different things for growing. And you can use cohorts, but you need a reference.
Yeah.
So I'm interested in the natural reproduction and recruitment of the next generations after you've done that, especially in places where maybe the population is extirpated. You don't know if you have the host fish.
Right.
Is there enough information coming back from looking at second generation?
Yeah, Virginia Tech has done a lot with, they probably had the first really long-term study, long-term funding, and long-term releases and replenishing panel, and they're able to go back and really get a good idea of that. And we're starting to too, in Norway and other areas where we're finding animals that we release, and we know that they're reproducing. Danielle?
Yeah, I just want to comment on that, and I was going to comment on that later, but since it's brought up, Catherine might hit me over the head, because I know there's traditional management paradigms, and then there's, I think, the future an ecosystem service-based future. We care about water quality and using shellfish for water quality. In southeast Pennsylvania, 95% of our streams that once held 6 species in abundance have no mussels, nor will they ever have reproductive populations. There's just too many dams on the streams blocking fishing. Would we not put mussels in there to get water quality benefits of mussels? If you can produce something like airway fluoride, so cheap, so prolific, reseeding every 10 years to get a return on that investment in water quality, I would just challenge us to not, we want natural reproduction to occur, but I wouldn't limit where we pick based on whether natural production biofishes. If I could just put that.
We still need to keep the mussel assemblage relatively consistent to historic. I mean, we don't
have
get it everywhere.
Asian plants.
Yeah.
And they felt their word too.
Yeah. Yeah, agreed. Yes.
Now, when I was looking at the it looks like you're planning to individual tags now. So there, see whether there's expectations they are going to move.
Oh, yes, they're gonna move.
So I wonder, do you guys have a general sense of how far they move away from your planting location? Like where you
That, Brian might be able to. You're smiling.
Some people have found their tag mussels over a mile away from where they initially lived. And
That's how many years though, like,
a year later.
okay.
That's not them moving themselves, that's them getting swept.
Right.
Right.
By
Yeah.
water.
Yeah. So, but TMD standpoint, that's something that needs to be considered. I'll talk a little more basically afternoon. I'm just taking notes. So I'm trying to relate what we do with oysters.
Right.
How they're similar to the freshwater mussel and how they're not similar.
Yeah, that's a good.
So, yeah, the planting, like usually they want to have a good sense of where you're doing the practice. That's where it's happening.
Okay, yeah, that's a good.
Something to think of.
You know, just, we could say though that, you know, the freshwater mussel beds have been well established in our rivers for thousands of years, and they're saying that
Right.
is still there. So while we can't ever account for them, if you're going to have high flood and flow, then it's going to take some downstream. I think
Those areas probably would be prime areas to kind of look at. If you're getting a consistent biomass of mussels.
And even then, they might go downstream, but they're gonna end up in the next suitable habitat downstream. So they're not gonna go all the way, they're not gonna be wasted or go away where they're not making a difference as far as water quality goes. So another thing that we do, we provide a lot of mussels to public agencies, nonprofit, universities, private entities. We provide a lot of mussels for toxicological studies and just culture method testing, getting new facilities off the ground, providing technical assistance, things of that. And last year, we distributed 20% of what we grew just for that cause in itself. So. And we have the capacity to produce literally millions, probably tens of millions of mussels a year. So I'm just going to talk about for a few minutes kind of what you need to grow mussels. You need a permanent funding stream. VF8OUC was started with collaboration between Fish and Wildlife Service and Game Department. And we can do a little bit here, and we've got mussels in a pole shed, we've got mussels in the carport, let's build another building. You can see this building right here was this is the first building that was constructed. So you can piecemeal things together. That does work. Very shortly, this building, we needed 3 times the size. So we built another building, and now we need a bigger building, you can piecemeal it together, but ideally, permanent funding operations are about $100,000 a year. I'd say $50,000 to $100,000 a year. We have probably by far the most staffed facility that I've ever had the pleasure of working with. And there's been years where I've been the only person. So we have 5 people now, which is, that's a good amount of money. That's over $350,000, counting my salary. 3 of those people are soft dollar funded, and you need at least a minimum of 3. And right now we have everybody out there has mussel experience and came from another mussel facility. So we're really fortunate, and we are, I would say, we're the best staffed facility, but I mean, in the big picture, it's a lot of facilities that are piecemealing things together. And so permanent funding for staff is always the hardest and most difficult challenge when you have a facility like this. No, it doesn't include salaries. Pumps, chillers, things on the next page. Yeah. Yeah. It's okay. Space, so 2,100 square feet is not out of the question. The next time somebody presents me with a plan, I'm just going to tell them to triple that, because you always end up needing more space. Things are growing, we're growing really quickly. And you need clean water source. I mean, we have the ponds, they have been incredible. You really need to have that, that's imperative. We teach a class on propagation. One of the first things I say to people is, have you had your water tested? Make sure that you have a clean water source. Yeah. They are lake fed. So they're gravity, we've got Harrison Lake, and then it's gravity fed. It's a creek, Herring Creek, where we get our blueback herring from. And partners, you need partners. Like there's no way this facility would be here if it weren't for Brian and Virginia Game and Fish, you know. So equipment, this is part of that, 100,000. When you're inside, you need to control your temperature. You've got chillers, you've got heaters, you've got pumps, you've got electric bills. There's so many things that you need to do this. Field gear, recirculating systems aren't cheap. So like I said, we piecemealed, we started out, we got mussels from White Sulphur Springs to start out, seeing if we could just grow them in the ponds, and then things just kind of slowly developed until we got where we are today. You need office space, lab buildings, all the infrastructure that the service provided, and ponds, of course. So is there a facility already somewhere, any available infrastructure, an old hatchery somewhere that isn't being used anymore that could be retrofitted? These are things that you always want to consider when you're thinking about starting freshwater mussel work. Always want to think about biosecurity or invasive species, broodstock, where are you going to hold them, how are you going to feed them? These questions always come up. And so it doesn't get done without people. These are, Michael Odom is the project leader at Harrison Lake, Brian Watson, you know, before, we've got Amy Maynard, Ben Davis, Bryce, John. I mean, we just can't do it without people. It's a tremendous amount of work. And with that, I will take any questions. Any more questions? Yes.
So I believe at the outset of your presentation, you suggested that Virginia has more mussel facilities than surrounding states, which started earlier. And I'm just interested in what that history was. What you started here in Virginia, why has it been successful?
Well, in Virginia, at first you have Virginia Tech. So you had Dr. Nubbs. You got a lot of research dollars coming in. So that facility is Hatchery University. That was probably the first facility in Virginia. And then the need for all of these upper Tennessee river systems. You had Big Hill, you had Lone Mountain, Mair settlements, Coal Pond, the dam broke and it was devastated out. It had been devastating for years, so you had a big influx of funding for that. That's what got the USFWS involved. Plus, the state had already been doing a lot of work since it's a state resource, state listed species. And then nobody was working in the area as well. There's very little going on, maybe except for North Carolina, in those species. Right now, there's 3 that are being petitioned to be listed in the Atlantic Slope on the eastern shore. So
So these animals are starting to decline pretty rapidly. And there's just no way working it, and that's what started with our slightly. So there's always a need. It's a good question.
So we're a little behind the time. So round of applause for Rachel. And if you have questions, please follow to the group lunch line. So just going to switch up the agenda a little bit for those that are early lunchers like myself. So we have lunch set up in the lab right through here. You just follow and you can go around. If you all can try to be back here in 10 minutes, we're going to have another presentation starting. Bring your lunch. You can eat over lunch, and we'll go ahead and kind of get going through. We might extend a little bit later into our Q&A session a little bit later. But thank you.
And so our next presenter is Jorge, comes from the Anacostia Watershed Society. So I had Jorge after we kind of set the agenda, so sorry to shove it over lunch, but Jorge's going to be talking about what their group has been doing in the Anacostia Watershed. And I felt like it might be just helpful. It's going to be a short presentation, but just a snapshot of what kind of small NGO is doing on mussel restoration in the Chesapeake Bay watershed. So take it away, Jorge.
All right. Yeah. So yeah, a little bit on the Anacostia River. Oh, this is not working.
Yeah, you might have to use the clicker. This is.
Oh yeah.
Okay. And left and right can go back.
Mm-hmm.
But this is a laser pointer right here.
Okay. Yeah. So yeah, we're the Anacostia Watershed Society. Our mission is to make the Anacostia River fishable and swimmable by 2025. You can see the watershed. It's mostly a Maryland river. 80% is on the Maryland side in Montgomery County, Prince George's County, and then the eastern half of DC makes about 15% of the watershed area. Okay, so this is the 2018 report card. So great news, we get a passing grade D. Sounds mediocre, but it's really great progress from 1989 when AWS was founded. As you can see, a lot of the water quality variables are trending to improve, with the exception of dissolved oxygens, which we don't know why quite yet. We have some theories, and then stormwater runoff, because there's a lot of development. This is a mostly urban watershed, so stormwater is still a big issue, as in many other places in the Chesapeake Bay watershed. Again, we did the mussel demo here, before and after. The tank on the left has mussels and the other one doesn't. So yeah, mussels improve water quality, and that's why we're working with them. And they help benefit other wildlife species and water quality. So what have we done so far? So we had no idea about mussels, besides some findings of shells here and there. But since we do a lot of wetland restoration work and a little bit of SAD propagation, we started seeing more mussels. So we got interested. So we partnered with the Maryland Department of Natural Resources. Not only in Maryland, we also work in DC with them, with Matt Ashton, the state mussel expert, and we did this work assessment of freshwater mussel community for the tidal Anacostia River, doing different kinds of samplings, including corner samples along the main stem of the river. And then after that, we have continued doing our own surveys up and down the river, with our interns and staff. And so to go back right here. So right now we have found 8 species of native freshwater mussels from the Mionidae family in the river, which is the same number of species found in the Potomac River, which is pretty good. We were surprised by that diversity. That's 8 of the 16 species found in the state of Maryland. So as we speak now, there's these small baskets, which are used for horticultural purposes, and then silos that have been used by the Fish and Wildlife Service, Fred Pinkney from the Chesapeake field office in Annapolis. So they deployed a bunch of baskets and silos throughout the river, and he's checking on survival and water quality around these cages. Before that, he did an ecotoxicology study that's still not published, almost ready to be published, with Eastern Elliptios, Elliptio complanata, and checking out different things, PCBs, heavy metals, and he found a great survival, like only like 1 or 3 dead mussels out of hundreds. So that's when the interest came in propagation. So we see great survival, so let's do the next step. Let's do this other study to see what's the survival using baskets and silos. So that's what Fred is doing, and he's using Eastern lab mussels raised by Rachel here at the Harrison Lake Hatchery. And then, so we're learning a lot from that, and of course we're partnering with Rachel Mair and the Harrison Lake Hatchery, and the next step is going to be our project. We are, with the help of Rachel, raising legumias, Eastern pond mussel, and also LY floaters for our own study. We're going to use these bigger baskets, fish baskets, and we're going to have them at 7 different sites throughout the tidal Anacostia River. Only 1 non-tidal site. Have you guys been to the Kenilworth Aquatic Gardens? So that's one of the sites in one of the ponds. The other are along the actual river in tidal environment. So these are the species that we have found so far. And you can see the conservation status, Maryland in light blue, and DC. So 6 of the 8 species have some sort of rank. Eastern land mussel, I guess, is status uncertain in Maryland, but as has been exposed today, they are very imperiled. There's a lot of species that are listed, and the ones that we are using is again LY floater and Eastern pond mussel. They're both listed. The SGCN is the ranking of species of greatest conservation need. So DC didn't actually have a good inventory of mussels because we did this work after they did the wildlife action plan. So they only listed these 2 species. We're using, 3 species, sorry, including the tidewater mucket. So we're excited to partner with the District Department of Energy and Environment to kind of like keep pushing mussel restoration going forward. And well, some of the abundance, we have found that the Eastern elliptios are very common, and Eastern floaters. And then you can see some of the less common species. And this is just some surveys we did last year on 2017. The Atlantic spike, tidewater mucket, and the paper pond shell are some of the least common. And a good sign is that they are reproducing. When we do the surveys, we found juveniles of small size like this Eastern pond mussel or this Eastern floater about the size of a quarter. And we've found gravid females and that's a great sign because there's other cases, you know, like in Oregon where they found this mussel bed and there's no signs of reproduction. So even though we have had so many challenges in the Anacostia, the future is promising. And you know, we even found them in our cages for submerged aquatic vegetation. One cage had all these. These are each cage is 100 square meters. So we found all these mussels, including juveniles, inside the cages. So next steps. So we hope to continue our partnership with the Harrison Lake Hatchery. More community engagement and education, because people don't know anything about mussels, but they like what they hear. I mean, they think we have oysters, and you know, that's the first step, educate them that we don't have oysters, we have freshwater mussels. And so far on social media, I have a display of the mussel species on a little board. We've been taking it to tabling events, and people is very, very interested. We're getting a lot of great feedback, not only from the public, but of course also from partners, from including the DC government, which is good. Maybe they will start funding our work in the future. And yeah, continued inventory and monitoring, and important is to determine the suitability and the long-term viability of release sites. You know, are they surviving, and then are they reproducing the ones we're releasing? So those are good questions going forward. So yeah, we're gonna have, so as we speak, Rachel has a lot of juvenile mussels of two species, again, LY floater and eastern pond mussels. We're gonna have them at 7 different sites along the tidal Anacostia, around 300 juveniles per basket for about a year, check for survival, average growth, and we're gonna release them into the river at different sites to be determined, because in the Anacostia River, there's an ongoing investigation on the legacy toxic sites and the pollutants in the sediment. So there might be some remedial action. We don't know where it's gonna happen and when, so we're kind of also depending on that a little bit, because we don't want to release mussels and then they're gonna get dredged or removed. And then monitoring, post-project monitoring, tagging using epoxy glue dots or something on the cheap side. If we get funding, we could do something great like what Rachel does with those tags. So that's the picture of Kenilworth Aquatic Gardens. So this pond right here, this is where we are gonna have some of our baskets. And right here is the tidewater muckhead, Leptodea ocratia, is one of the species that are found in the Anacostia River. It likes tidal freshwater systems, so Anacostia River is a good site for that species. And then community involvement and education. We're excited to do the Mussels in the Classroom program, which has been modeled after the program by the Ohio River Foundation, which they do in partnership with the Kentucky DNR. So it's going to be similar to our other programs. It's a 3-part classroom, and then boat tour and field experience with mussels. The kids are going to be able to raise mussels in a 10-gallon tank in the classroom, you know, like 10 mussels per tank, and you know, keep them learn about their biology and ecosystem services. And then we're going to put them into the baskets in the river and then eventually release them. It's a pilot project. We're going to start it this coming winter, and we're going to have 5 schools and we hope to engage about 250 students. So yeah, we have similar programs like with wetland plants, like the Rice Rangers or the Shad program, where kids release the shad fry. So this is a new one for us, and we're very excited. There's also a lot of schools that are very interested already. So yeah, people also we're gonna have some volunteer opportunities with maintenance of the cages and monitoring. Maybe we're thinking even some citizen science component, and then the student experiences. All right, and we have started to use this hashtag Mussel Power. So yeah, thank you.
So we got a round of applause for Jorge. Maybe time for one or two questions just while we're changing out the PowerPoints here.
Sorry, my mouth is full. Is there a time of year when the baskets have to come out of the water? In other words, what is the growing season that you all when the kids go from the basket?
Yeah, so they are going to be from, let's say we're going to deploy them in August, so it's going to be until next year. We're, they should be able to survive the winter. Baskets are deep enough that they're not going to freeze over. Although that could be a risk if it gets extremely cold, like we did at the beginning of this year. And another thing is that we're testing baskets for tidal sites, which are mostly pretty, stagnant, but there's flooding events of recon, so we're gonna have to keep a close eye during and after flooding events. So that would be the main risk. But normally we're gonna leave them in all that period from August to August, in the year.
So they're weighted down somehow, so they're not.
Yeah, we put a cinder block, or we put some structure like a PVC pipe frame to keep them in, and also galvanized fencing to keep the turtles out and other critters. Yeah.
Thank you, Jorge.
Yeah.
So our next presenter, Danielle Kreeger, an old colleague of mine, we were fortunate to serve on the Atlantic Estuary Research Society board several years ago. She was the president of that. And she comes to us from the Partnership for Delaware Estuary, our sister estuary, if you will. And she's going to be talking today, some really cool stuff going on up in the Delaware Estuary. But she's going to be talking about these ecosystem services we talked about. And specifically, this I think, this is right, Danielle, comes as a result of the 2010 Obama executive order. That there was something in that executive order that requested an investigation of whether mussels can play a role in addressing pollution loads. And I think Danielle with her colleagues has been doing a lot of the work to finally get an answer to that question. So take it away, Danielle.
Thanks, Joe. And I really am grateful to be here for many different reasons. First off, I just love talking about mussels. But also we do have an agenda, and one of the agenda items is to do more work jointly between the Delaware Estuary and the Chesapeake. Common set of assemblage is from the mussel taxa side, but also shellfish, and we tend to well, there's a great brain trust in the Chesapeake and a great array of resource agencies really focused on very similar questions, and over in the Delaware system, many of those questions are the same ones, but we don't have the resources, we don't have quite the brain trust. I'm really looking forward to building bridges. I spent time at Horn Point, and there's some Horn Point people here. I came from the marine world. I was trained as a marine biologist, and as I mentioned during introductions, Catherine Gayton connected me up with Dick Neves, and I was on her committee. That's how I got interested in mussels. I still do the marine stuff too, but I will warn you all, if you haven't already become a mussel head, I have lots of people that have come to me as fisheries people, water quality people, and they became mussel converts pretty quickly. And I give talks on, climate change and wetland ecology, and I teach, and it's been always really astounding to me that some of the best reactions I get, some of the most invigorating after talk discussions and people following up afterwards have been after the mussel talks. And I think there is this, what Joe started off the day talking about, the opportunity for engagement up in the watershed, that's definitely true. But there's also something really sexy about these animals. I mean, that cool life history strategy, and just the fact that, people just don't know. It's like there's so many did you knows. You know, the world's epicenter for unionic biodiversity is here. The fact that they live to be like old growth trees and streams. And if you're, as someone was talking about indicators, we use freshwater mussels now as one of our estuary program indicators. But the whole field of stream ecology, the freshwater stream ecology, aquatic ecology evolved from entomologists that really did not give fair due to the long-lived sentinel bioindicators of stream systems, right? Whereas in marine biology, the field of marine biology partly evolved from oyster shellfish biologists and fisheries people. And so there was a really early and long history of research on ecosystem services in the marine bio world. But translating that over to the freshwater aquatic ecology world has only happened very recently. It's only happening now. So the Freshwater Mollusk Conservation Society, which was really created 30 years ago, when I went to my first FMCS meeting and I gave a talk on ecosystem services in the late 90s, I was virtually run out of the room. I mean, it's mostly systematics folks interested in saving the rare species. And of course, we're really interested in conservation biology. But as a national estuary program like the Chesapeake Bay program, the Partnership for the Delaware Estuary, we're affiliated with the Clean Water Act, not the Endangered Species Act. So for us, it is water quality. And what we do at the estuary program is we really have an ecosystem service-based goal structure. You know, where do we get our carbon sequestration? Where do we get our nutrient removal? That kind of stuff. So we're focused on these animals at the estuary program, mainly for water quality reasons. However, close second is the rare species. So I have a bit of a talk. I'd like to run through it and then do questions afterwards. If there's a burning question as I go through, please stop me. And I do want to get at this. As Joe said, you know, I've been working on ecosystem services. That's my career research, freshwater and marine. But this idea of connecting it to management goals, you know, is something that's really come up in the last 10 years. And it's really, I think we could see this coming years ago. To me, it's absolutely is the future. How quickly we get to that future with BMPs and all that, you know, remains to be determined, but it absolutely is the future. Just like carbon markets are coming back online with Reggie, and in Europe, they're building mussel farms, marine mussel farms, to sell credits on nutrient trading markets. You know, that is the future. That is where we're headed. But we do need a lot more research to be done. And the one thing I want to start off with, and it's my big caveat. Number one, we're not trying to take anything away from oyster restoration. Okay? PDE, our organization, we just like the program here in the Oyster Recovery Partnership, all the good work that gets done. Oysters are really foundational species, and we are deeply invested in oyster restoration too. But oysters live in one sort of niche in the system. You know, they're not everywhere. They certainly aren't up in the non-tidal freshwater tributaries, right? So, and second thing is we're not trying to take anything away from all those other great BMPs that are still needed. You know, stormwater controls and keeping the cattle out of streams and, you know, riparian quarters, all that stuff, so critical. In fact, there's many streams where I don't think we can restore mussels to yet until we fix some of those stormwater issues. I mean, mussels need habitat. Mussels are patchy just like oysters. They need habitat, and a lot of our streams are just ripped. And you can't grow mussels on bedrock, right? So anyway, those are my two caveats. I had to get them out first. So again, oysters, you know, a long 100-year history of ecology and biology, and where I got my start on nutritional physiological ecology. And in Delaware Bay, those are some of the oyster reefs. Interestingly enough, with climate change, one of the upsides of climate change is that we now can have intertidal oysters in Delaware Bay. And that's a niche expansion for us, and we can do living shorelines and all this cool stuff. My work measuring their clearance rates and so forth, I mean, there's different numbers you can use, but my work shows that based on existing standing stocks of oysters in Delaware Bay, they're filtering about 11 billion liters of water per hour. The only reason I put that up there is, well, they're not the kingpin. I'm going to talk about freshwater mussels in this talk, but I have to also say, please don't forget red mussels, the marine species either. In Delaware Bay, where this is the salt marsh mussel, they live dense in the intertidal zone around marshes. We have a lot of wetlands in Delaware Bay, so we have a lot of red mussels. So if you're doing this black box exercise, it looks like red mussels are filtering probably 6 times more than oysters in Delaware Bay. So of course, we have oysters and red mussels, and we're going to be talking about these other species. And so I put on here the Chesapeake and the Delaware River, watersheds, because I tend to think of them as a whole. I care about both. And for the freshwater mussels and the marine species, we're generally dealing with the same suite of taxa. Fewer of the freshwater mussel species like the eastern spike, we don't have those in the Delaware River Basin, but the 12, 13 species we have are also in the Chesapeake, and they're the functional dominants of the assemblage. So anyway, so there's actually about 60 species of bivalves living in these watersheds from headwaters to the ocean. And some of those are not really that abundant, but some of them historically were. And just like oyster reefs provide complex three-dimensional habitat structure, ecosystem engineers, not many people know that a natural healthy mussel bed can reach very high abundances. The historical records have all that flowery anecdotal stuff, but we can go to there aren't many great reference streams left, some in the West where I've worked with tribes, but we have mussel densities even in the Delaware River Basin in certain places, 100 to 150 per square meter, and these are big animals filtering a lot of water. So I'll show you some data for that in a minute. Anyway, so oyster reefs habitat, mussel beds habitat, there's all these other benefits. If you look at the Millennium Ecosystems Assessment, from 2000, and how they categorize things, these shellfish, freshwater and saltwater, are providing many different benefits. And today we're going to be talking about water quality, but just a reminder, here we have oysters, red mussels, and freshwater mussels, and these are just different ecosystem services. There are many ecosystem services. Now, oysters being commercially valuable, they've got 3 checks here in this column, but, red mussels, freshwater mussels, generally not commercially important. But look at, biofiltration. My point here is, well, all of them do this. And I'm going to show you some data to confirm that. One of the long, paradigms, and I don't know where it came from, is, oysters are like the kingpin for water filtration on a per animal or per body size basis. And freshwater mussels, longer lived, slower growing, probably not filtering as much water per animal. That was really a myth, and we're going to bust that myth. So here's what we're talking about. We saw it before. We have Brian's slides, the water filtration demo. If I have time at the end, I can show you that on our mightymussel.com website. We have a time-lapse video. It goes through about 2 hours and just shows this right before your eyes. We do this at all of our outreach events. If we're doing an outreach, an environmental fair in the Bay, we use oysters or red mussels. If we're up in Philly, we're doing freshwater mussels. Same thing. And it's nothing makes the case better. We just did Philly Science Festival, and we had a competition. Who filters faster, and oysters or freshwater mussels? So we put the same, because it's all about experiments, and so we had 4 tanks instead of 2. We had saltwater and freshwater. And then we were using little baby Secchi discs to track turbidity over time with all the kids. We say, come back every half hour, and we get data on the big flip charts. It's great for outreach, and they get it immediately. All right, now the bad news, of course, is that all of these native bivalves, headwaters to the ocean, have been in decline, are still in decline, are threatened. Freshwater mussels, Brian summed that up nicely. Oysters, of course, with sea level rise, warmer, saltier water, prone to diseases like warmer, saltier water. There's a bit of an uphill battle there, and there's some other issues. Red mussels, the kingpin in Delaware Bay, we're losing an acre of marsh per day. An acre per day loss rate of salt marsh. So their habitat's washing out from under them. And I get a little frustrated that we hear some of these great projects are great ideas. Well, let's build a red mussel farm in the Bronx and all this stuff, and let's put some money into that. And you spend a lot of money and you create this little island of maybe ecosystem services for high value. But at the back door, no one's paying attention that you're losing 20, 100 times that amount of biomass every day, when a simple living shoreline at low cost could have probably stemmed the bleeding. So we need to think of what we can do to increase the water quality benefits of shellfish, but don't lose sight of the conservation need. It all starts with conservation. We do have threatened native freshwater mussels and marine species that need to be first protected, and then we can work to enhance or restore. So that's the bad news, but the good news is we now have the technology and we're learning more every day. Rachel and Brian have been truly taking the baton from Dick Nebbs and really moving things forward here, as well as others, Chris Barnhart down in Mississippi. And there's some really great research on the freshwater mussel side on propagation. And of course, we know we have to keep taking out the dams. There's habitat considerations. We need to promote an ecology that supports these species. We have been working with the marine species on all these new types of living shorelines, and now we're moving into Philly and Camden and Wilmington with freshwater living shorelines that should have freshwater mussel beds. So this is all coming online. We're developing this new green tech to increase this natural infrastructure to provide ecosystem services. And it's exciting, and I wish I was young and starting off. I'm jealous of people like that. You know, because there's like so much cool stuff coming. But it begs a question, if you're a manager and you've got $1 million to invest and you want to get the nitrogen removal in your watershed, TMDL or whatever, what tactic, what species, where, what's the return on the investment, right? So this is what we need to start thinking about and being honest about. So if you're interested in how these species stack up, and there's a lot more than what's on the list, but these are some of the big considerations. Physiological capacity, you know, does an oyster filter more water per pound biomass than freshwater mussels? Who does more? What was seasonality, different life history stages, and so forth. And then what is the current and potential future carrying capacity for populations of those animals? These animals all live in different niches. They all have different habitat requirements. Some of those habitat requirements are going to be tough, you know, to fix or enhance, and some might be totally, there's great opportunities now. So we need to identify where we can get an increase in population biomass, which is how ecosystem services are scaled. We need to be honest about ecological barriers. So the diseases of oysters, the dermo and MSX is an example of an ecological battle, you know. There are ecological considerations with freshwater mussels. The fish hosts are the first one. Do you have the fish hosts available to get natural propagation? Not necessarily a barrier if you're really just trying to manage water. As I said, just reseeding every 10 years, if you can do that cheaply enough. I mean, we couldn't take for trout fishermen, right? Why couldn't you couldn't take for water quality benefits if it's nominal cost? These animals still live to be a long age. So if you can get them into your system and not kill them again with another spill or something, then maybe they can perform these ecosystem services for you for free for a long time, and their services will increase in a non-linear fashion as their biomass increases. So even if you don't get the benefit of natural progeny, it still might make sense. Policy barriers, well, in New Jersey, we can't restore oysters everywhere. If the waters are closed to shellfish harvest, we can't restore oysters there because of the worry about poachers poaching your restoration project, getting sick, and it's a liability issue. Not everywhere, you know, but New Jersey, that's an example of a policy barrier. Well, with freshwater mussels, there's going to be policy barriers. And the big traditional management paradigm with all these species, marine and freshwater, is to restore to historical conditions in historical places. But guess what? Climate change, everything's on the move. So I mentioned about oysters now intertidally. I don't know what New Jersey's going to do once oysters start growing intertidally in all those places where the water quality stinks. they're gonna have an eradication program. Freshwater mussels, you know, we have species that we care deeply about. Eastern pearl shell. It's a cold water species. It has a brook trout as its fish house. And right now in my area, the only place you find margaritifera is below dams that have cold water bottom release. Is that going to be a big restoration target for us when they're probably not viable in the future in this area? What about the southern species that might do well here in the future? As niches open up, are we going to let them just stay open, or do we have to think about the blasphemous idea of assisted migration? Because these freshwater mussels that disperse on fish hosts that are freshwater fish are not going to be able to redisperse northward from southern populations, whereas marine species that are broadcast spawners easily disperse. Right now you got blue crabs, fishermen replacing lobster fishermen in Long Island. I mean, everything's on the move. And the marine species are going to be just fine. The freshwater species that require freshwater fish, how are they going to get north?
How do fish?
Huh?
How do fish?
How do the fish get north? When there's a saltwater barrier. So those are the kinds of things what's new. And then willingness to pay. Oysters, everyone loves oysters. I love oysters. I love shucking oysters, eating oysters. But, and there's a bit, you know, obviously there's a charismatic, you know, everyone wants oysters. But freshwater mussels, I'm optimistic that with increasing outreach programming and people getting the word that there is going to be a lot more caring about these animals, the mascot Joe talked about. So hopefully willingness to pay will increase. Anyway, but these are different considerations, and I want to focus now a little bit on the physiological capacity because that's where a lot of my research has been. And Joe really asked me to sort of summarize the literature. And as you mentioned, the Chesapeake, the executive order for the Bay cleanup basically had this FW20 white paper as an action item or a task to evaluate native bivalve restoration for water quality improvement. Of course, that includes oysters. But then action 1, complete a literature review of relevant studies on the ability of tidal and non-tidal freshwater and estuarine bivalves to enhance water quality. So Peter Bergstrom was a NOAA, I forget what his position was at NOAA, scientist at Annapolis, and he had reached out to me because he saw some of the stuff we were talking about, and Catherine Gatenby, I think, was involved in getting this on the list somewhere early on, right? Anyway, so we decided, well, let's tackle that. Let's tackle this white paper, sort of a research review, really looking at it from the physiological capacity side. And so we started working on this paper. God, I hate to. 2013. Anyway, there's been various drafts, and we just finally, Peter retired, and so Catherine and I are finishing this. We're almost done, hopefully the next month, to submit it to a journal. But it basically looks at marine species of Chesapeake Bay and Delaware Bay, and the freshwater unionids, and also considers, just because we got to consider the non-native species, you know, and not that they would ever be a target for restoration of non-native species like Asian clams, but they're there, and you have to recognize that they do filter water too. So what do we know about their clearance rates? What do we know about their filtration rates? What do we know about their restoration promise? What's the state of the technology? So this paper will be available soon. And there's all these tables in it because it's a review, but I just want to point out a couple things. Clearance rates, and this is just the start of some of the saltwater species, and it's not totally comprehensive, all the literature that's ever been done on oysters. That would be a whole book. But the one thing I want to point out is in red here, lab diets versus natural diets. If you're looking at the water quality benefits of any of these species, freshwater or marine, are you going to be feeding them a lab culture of the xenic algae and then actually translating that to what that means in Chesapeake Bay? You know, where they're feeding on a rich soup of microparticulate material that includes bacteria and detritus and all this junk. Most of the research that's been published on these species, both freshwater and marine, comes from studies that are interested in captive care or aquaculture. How do we grow oysters fastest with the best possible diet we can grow in a lab? if you get into ecosystem service studies and water quality benefits of these shellfish, you have to be looking at natural diets, right? And so Joe, you've done some work on this too. So this paper really differentiates that a little bit, and from a water quality standpoint, I think we really should be thinking about natural diets. So then I was trying to say, for this talk, you know, and for this white paper, what data do we have that's intercomparable? Because the other problem is people just use all these different crazy units, and they don't scale their measures of clearance rates to the body size of the animals well enough, the dry tissue mass of the animals. They'll just say per animal, per clam, liters per clam per hour. Well, how big was that clam? Right? And so anyway, so this talks about that. What I want you to get is that from an oyster's perspective or freshwater mussels perspective, they're not trying to improve the water quality of Chesapeake Bay. They're just trying to make their ends met. They're trying to meet their nutritional demands. The nutritional demands of these animals vary widely throughout the year. Are they trying to build up energy reserves to get through the winter? Are they trying to take on a lot of protein to fuel gametogenesis for reproduction? So the energy requirements, the essential biochemical requirements vary throughout the year. They also vary throughout age, because young juvenile mussels are growing very quickly. It's all about biosynthesis. It's a very high protein demand. As you get closer to reproductive age, and then you start to think about lipids and other things. So there's a lot of variability in what they need. At the same time, food quantity and quality, that soup of particulate material out there in the James, I guarantee you it's different than it was a week ago, right? TSS is way up now, right? And the chlorophyll and the protein content and all that is going to be much changed. So and there's a lot of seasonal variability, spatial variability. So that soup is constantly changing, and their nutritional demands are changing. And so rarely will what's in the water column be matching their nutritional demands, and they have to compensate. Physiologically, bivalves are some of the most incredible animals for adaptivity, an optimal foraging strategy, but it doesn't come at the clearance phase. At the clearance, just sweeping particles from the water column, it's fairly conservative. In other words, it's the energetic costs associated with filtering particles, the steam of the cilia and all that stuff, are very, very low in an energy budget of an oyster or a mussel. The high costs, the energetic costs, are in the gut, producing those digestive enzymes. Therefore, they are almost always feeding maximally. It is fairly consistent, their clearance rates. But what they do with particles in the gut, there's 20 different places where they can sort, select, and maximize enzyme substrate efficiencies. And so the reason why I'm bringing all that in here is because we can model the water clearing benefits of these animals pretty well. But we should be honest that what comes out the butt end of these animals, how much they feed back to the water column, all this the net water quality benefits is going to be much more variable than the gross renewable part. So I'll come back to that in a minute. All right, so as I was trying to put some bars together to compare species, I really realized during this white paper effort, it gave me a chance to reflect on my career. So over 30 years, I've used the exact same methodology. Well, I've used a lot of different methods to do a lot of different things, but for answering these questions about water quality, I've used the same method of measuring clearance rate with oysters, with rib mussels, I had a student do Asian clams and all these different freshwater mussels. And I thought, well, that's actually the most comparable data set I know comparing freshwater to marine. And so I just thought for this, I would show you. So I have data for this clearance rate data for the Pacific slope. This is out in working with tribes in the Pacific Northwest. Catherine and I did a lot of work in the Mississippi Basin over in the Allegheny River with freshwater mussels. And then Catherine and I also did work with Elliptia complanata up in the Brandywine Creek area. I have a student, Kurt Chang, who did work with Asian clams, clearance rates. And then my early work, plus my recent PhD student, Josh Moody, did a lot of work with rib mussels, and I also had data for oysters. So this is my only data slide. Okay, so, and some people in the room aren't scientists, but bear with me. On the y-axis here are clearance rates, and this is liters per hour per gram dry tissue weight. Okay, so they're scaled, and these are allometrically determined. You know, so there's all different sized animals in these experiments to actually capture the allometry, but this is normalized for weight. And then these are just different species, and the dark blue bars here are different species of freshwater mussels from across the United States. And the red is Asian clams, and the green is rib mussels. This is for, and all I did was all these experiments, I used the same method. I used natural cestine diets that were in the water column where the animals were taken, natural temperatures. But I just, to do this comparison, I said, well, let's just filter the data, let's stratify the data for everything that was just over 20 degrees, summertime temps. Okay, so you have summertime temps, everything normalized for dry tissue body mass. And what you see here is all the bars are actually fitting on the same screen. And so these are standard errors, and those are high error bars. I mean, bivalve clearance rates, anybody who's done physiology work with shellfish knows that they're notoriously variable one individual to the next. So there's a lot of variability, and you might say, oh, I'm not sure if that's significantly different from that. It doesn't matter. My point here is that over all these species across the United States, freshwater, brackish, marine, 1/2 liter to 1.5 liters per gram per dry tissue weight. Now those numbers might be lower than what you've seen in the oyster literature, 5, 7 liters. But again, a lot of those studies weren't with natural diets. If you look at this with natural cestine, natural diets, generally the rates are a little lower. Let's be honest about that. But my point here is freshwater mussels, the blue, they're no slouches. No slouches. And then if we filter all those experiments, 15 to 20 degrees. This is sort of late spring, early fall. Now you have oysters on there. Oops, oysters aren't, the bar isn't any bigger than the others. Oops. They're just pound for pound. These guys are, you normalize for body weight, normalize for temperature, using natural diets that are incredibly variable, which gives you some of this variability. The clearance rates are fairly consistent. Clearance rates. That is the volume of water swept clear of particles per unit time, per unit body mass. What is a filtration rate? Okay? Filtration rate means you take that volume of water and what were the particle loads in that water, the mass of particles in that water, convert it to a filtration rate. So then we have to say, okay, what were they feeding on? I have one more data slide, sorry. So those same experiments, each dot here is an experiment that I've done sometime in the last 30 years with colleagues all across the country. TSS is the blue squares, just % organic content of the cestine is the red circles. And it's like a shotgun pattern. My point here is that what they're feeding on that soup of material is so variable. It's ranging from maybe 5 to 150 milligrams per liter TSS. And from maybe 10% to greater than 80% organic content. So the quantity and quality is highly variable. So then you multiply like TSS times the clearance rate, and oops, now the variability gets completely wild. 1 to 314 milligrams filtered per hour per gram by tissue weight. And my point here is you really need to know what they're filtering. If you take an oyster and put it in the James versus an oyster and put it, I don't know, find on the Lake of Trophic area, an area that is fairly clear water, who's going to provide the most ecosystem services? Well, it depends what you're really interested in, but I can tell you who's going to filter more cestine, and if there's more nitrogen in that cestine, the nitrogen removal, the water quality benefits are going to be much higher if they're feeding on material that's more nitrogen rich, right? But no one ever really looks at that much. We talk about clearance rates and that's all the end of the story. You really need to talk about filtration rates. You really need those data for what they're feeding on. All right, so I'm going to sum up this part and then move on to another point. Clearance rates of water were comparable among freshwater, brackish, and marine species. Freshwater mussels are not slouches when normalized for body size and temperature, but the filtration rates of particles and associated pollutants, will depend largely on that composition of that material. So what this means, I think, is good news for modelers is there's not a big species difference. If you do work with oysters, you can take those models and really relate them pretty easily to freshwater mussels. You need to know what? You need to know the population body size, the size class distribution, and the abundance of animals that you have or who have. You need to know what they're feeding on, assessed on, the particulate nitrogen, particulate phosphorus. They're not going to help you with dissolved nutrients. It's particulate phase, 2 microns and up. You need to know the temperature, because these are cold-blooded animals, so it's a seasonally relevant variable. And yeah, so that's it. So what does this mean? So just if you take that organismal data, those clearance rates for gram dry tissue weight, in the Delaware River, we've done quantitative freshwater mussel surveys at 4 of what we consider reference beds. These are just upstream from Philly, between Philly and Trenton. Now, most of this shoreline is heavily altered, heavily degraded. Mussels aren't in many places, but where we had mussels, we decided to survey them and say, what are they doing for water quality, right? With the idea that, well, if it's a big number, maybe we could expand those populations and get more ecosystem services. So at these 4 sites, they cover about 7.2 hectares. We surveyed, there's 2.4 million animals, freshwater mussels of different species. There's about 5 species in this assemblage. And using just a seasonally normalized clearance rate for the taxa, we have a lot of new data in the last 2 years, 4 species on clearance rates, filtration rates up there, New Jersey, Pennsylvania, Delaware data. So that's all coming. This was just a simple proxy. But we're estimating that that bed of 2.4 million, those 4 beds of 2.4 million animals are filtering about 10 million gallons of water a day. And based on the TSS and the water column up there, it rolls up to about 10 tons of dry TSS per hectare per year. Now, again, there was only like 4 places that we could find mussels. Philadelphia Water Department is a big partner in this project. They take 400 million gallons of water per day from the river for drinking water for the city of Philadelphia. How many hectares of mussel beds will we need to get pre-filtration and cut down on their mechanical costs of water filtration, like Brita filtering the water as it comes in? So there's the wastewater treatment options, but then there's also drinking water pre-treatment options. They're interested seriously in trying to rebuild mussel beds near the drinking water into the city of Philadelphia. Now, on that bed, knowing the particulate nitrogen in the water column and that sort of density of animals, it works out to be about 423 pounds of nitrogen per acre. Sorry, I switched units, hectare to acre. So it's about 1,500 pounds of nitrogen per hectare per year on those mussel beds, just doing what they normally do. And they have to do that to meet their nutritional demands, okay? They're not doing it, but this is all gross removal. Remember what I said, gross versus net. Again, the physiology comes into it. We're talking about research needs for the future. We're getting really good at measuring clearance rates and so forth. We need the biogeochemists. We need the people who really understand chemistry to help us think about the returns, the fate of the filtered material. So there's no doubt they're filtering the water. They're increasing light penetration. If you're an SAB bottom plant, good, all good. Gross removal is all you probably care about. But if you're a water quality manager and you're interested in how much of that filtered nitrogen actually gets returned to the system, the fate of the filtered matter, well, you're going to have different pulses come out. The first is the P, the ammonia. Ammonia gets returned to the system. You know, they're going to pee some of that out. Then they're going to poop some of that out. And then eventually the tissues will turn over. Now, some of that will get buried, you know, the shells, the mussel tissue, and some of the defecated material will get buried. So you have a burial net loss. You also have the denitrification, potential denitrification within the beds, and that's going to be higher where you have denser mussel assemblages. So much more work needs to be done on denitrification, just like we have new data coming for oyster reefs, and that's all factoring in the oyster BMP thing. That all needs to be done for freshwater mussels. I have data, we have data on the ammonia excretion rates, and I've got a slide I want to show you here that's just food for thought. And then, again, this is just being honest. This is not freshwater mussel, this is a saltwater mussel, Metallus edulis, the restaurant mussel, yay. And this is O to N ratio. What this is the ratio of oxygen consumption to nitrogen, ammonia nitrogen excretion. And this is on the x-axis, basically the nitrogen content of what they're feeding on. So if you're think of this over here to the right as a eutrophic, nutrient-impaired estuary. Think of this over here as a site that's more nutrient poor, cleaner water. Animals that are feeding on nitrogen poor resources are going to be nitrogen starved, particularly when they have higher protein demands. Therefore, the O to M ratio is higher, meaning that they're producing less ammonia. But an animal that lives in a eutrophic estuary, it doesn't need to conserve, it doesn't need to spare its protein from catabolism. It holds on, or it releases a lot of that ammonia. So this is like they're sponging up and holding on to their nitrogen, and this is where they don't need to anymore, so they feed out. So what that means is your ecosystem services, your net nitrogen retention in a polluted nitrogen-rich estuary, you might have lower services, but if you can have a lot of animals, it still might be a huge, big number. Okay, so don't let that be a downer. It just means that our modelers, we need to know some of these physiological data to be realistic in terms of gross versus net nitrogen removal. So I wanted to just, another sort of caveats, varies. All right, so absorption and excretion rates depend mainly on body size, nutritional status, just like before, but in food quality and quantity. So their gross filtration doesn't always mean net nutrient pollutant removal to the system. So that's where I'm going to stop with the whole physiology story. I went a little deep into that, but I wanted to, I know there's people here who I think will, be asking those kinds of questions. Well, what do we know about denitrification? Nothing. Yet, I had some grant proposals that never got funded. So anyway, so at PDE, and what we think is right for the Chesapeake Bay, which is part of the story for this white paper we're working on, is that we should really be thinking of a holistic shellfish restoration strategy for the watershed. Headwaters to ocean. Non-tidal, intertidal, subtidal. Because these species are patchy. They live in different niches, even within the freshwater mussel world. There's riffle species and big, deep water, thinner shelled species. They live in different places. So if you want to get the best water quality bang for the buck, by all means, do not focus on a single species. Look at the places you want to remediate, what species did or could live there. That's the way I think might make the most sense. Now, just to finish up, I wanted to play around with some of this ROI stuff. Just starting to think about this. Where do you invest? There's the places, and you have the Anacostia or whatever, you have places, you're going to focus on, so you're going to know what species to work with. But if you're thinking of the whole watershed, should you really put all your efforts into freshwater mussels up in some headwater streams, or should you really stay with the oysters down in the bay, or rib mussels and living shorelines? So just playing around with this. These airway floaters, which is one of the dominant species we have in the Delaware River, and we've heard a lot about airway floaters here. This is the population biomass at one of our sites on the Delaware River, where we've mapped the biomass. They can be down to 40 feet, by the way. And they can be 1 foot below mean low water to 40 feet. And they're like small footballs. So they get big, they filter a lot of water. And we're tinkering with picking up on what Brian and Rachel are doing here. At least, they don't have, we've already tried, they don't have the capacity to give us our millions of mussels per year, and we want to go big on this. State of Pennsylvania is right now committed funding for construction of a mussel hatchery in Philly that would provide seed for the Delaware and the Susquehanna River, and we're looking for partners. We don't have it deal inked yet, but we're taking around, if this hatchery can produce 500,000 airway floaters per year, and you just saw some numbers from Brian, that's totally doable because they're the more weedy. And if we, stop those mussels into sites that were more nitrogen impaired, and we make some assumptions about their survival, their lifespan, and so forth, and we've estimated very similar to what I heard Rachel say, about $400,000 per year for the cost for operations and maintenance, then we'd be producing mussel seed, somewhere around $1 per mussel for airway floaters. It'll be much higher for species that are trickier, tougher, but airway floaters are easy. They're kind of weeds. We've learned that from these guys down here. what would you get? Well, over first 16 years of running this program, producing those seed, putting them in streams where you have impaired, per year we're estimating TSS would be, you know, somewhere around 1,000 tons removed. From that investment, you would get a return of about, this considers mortality and all that stuff, 1,000 tons removed. Oh, and it assumes no propagation. You might get babies coming off these mussels, but then it would even be higher. So this is somewhat conservative for that. And we've been looking at a 30-year timeline for what we're calling the Mussels for Clean Water Initiative. So that would then generate over 30,000 tons of TSS removal. Okay, well that's kind of cool. But what about what you all really want to know about, which is nitrogen, right? So this is all just a black box estimate, you know, so there's not a lot of modeling rigor. So this is only for discussion purposes, please. Only for discussion purposes. Don't tell your congressman or whatever, right? But if you're producing 500,000 alewife floaters per year, some die, some you put them in impaired waters, and then you get this, the net removal, you know, through some of our estimates for denitrification and burial and all that, you'd be getting about 78,000 pounds of nitrogen removed per year by year 30. So there's this nonlinear increase because those animals that you seed in year 4 or 5 are going to grow. Some will die, so there's a mortality term in here, but then they keep, the ones that survive keep growing, so the population biomass keeps growing. And if you then, this is per year, and if you look at the cumulative nitrogen removal over the lifespan of that project, you know, somewhere be closing in on 1 million pounds of nitrogen removed over a 30-year timeline. So so that's 400 pounds of nitrogen per hectare per year. TSS would cost about $400 per ton. Nitrogen removal would be about $14 per pound. How does that compare to other BMPs? Some of you in the room know that. What I've heard, you know, for like riparian projects and other things, you're looking at $100 to $200 per pound of nitrogen, something like that. So this is, so the state of Pennsylvania saw this, and I work with economists, and they're like, well, okay, so Pennsylvania is really still the laggard with regard to nitrogen removal in the Bay program, and there's a lot of pressure on the state to do more, you know, and so they feel that they've picked a lot of the low-hanging fruit. They're looking at maybe going to the next tier of more risky projects, and this would be a risk. No one's ever done this. We don't know if you're going to get those benefits or not. But the fact that you're an order of magnitude better than other BMPs in your first estimates says it's probably worth the gamble. So the state of Pennsylvania is looking to pitch in and try to help us get off the ground, at least with construction. For operations and maintenance and all that, we're looking for partners. And this is Susquehanna, Chesapeake, and Delaware. So please, if you're interested in this, talk to me later, because we're really looking to 4 or 5 years from now, what can we do to really operationalize it. So how does that compare to other tactics? I was going to do oysters, but I didn't want to, I thought that could be risky. So I did rib mussels because we have data for this. So rib mussels in these salt marshes, we know their biomass, we've mapped them, we've done clearance rate and nitrogen removal studies out the yin-yang. And so we know for this species, they're removing about, on average, it's again highly variable. For a healthy marsh that has a lot of rib mussels, you're getting about 92 metric tons of TSS removal per year, which, by the way, is enough to contribute materially to marshes keeping pace with sea level rise. It translates to about 5 millimeters per year if all that mussel poop stayed in place, but it doesn't. You know, some of that washes out, but it's not inconsequential. But from a nitrogen standpoint, it's about 1,000 pounds of nitrogen per hectare per year that they're pulling out and putting into the sediment. All right. The trouble is, as I mentioned, we're losing an acre per day. So we're seeing a daily decline of less filtration capacity in Delaware Bay of about 8 million liters per day less filtered because of marsh loss. So we've been working a lot for about 10 years on various types of living shorelines with Rutgers and other groups, developing new ways to promote oysters in the low intertidal, rib mussels in the mid-high intertidal, to stem the loss of marsh, but also to boost water quality benefits. And a lot of our funding for living shorelines is water quality. State of Delaware, and other 319 type grants from New Jersey. So there's a lot of interest in this. But what does it cost? Well, a healthy mussel bed, again, 1,000 pounds of nitrogen per hectare per year. A typical bio-based living shoreline like this is somewhere $10 to $200 a foot, depending on how you build it. We're just assuming for this 100, you can shoot the middle. Then your nitrogen services cost about $31 per pound. Not quite as good as the freshwater mussels, but still competitive compared to other investments you might make. And of course, there's all kinds of variability here. So this is really just for discussion purposes only. So for this return on investment, freshwater mussels looks good. Rib mussels looks good too. And, but we should also think of the decline trajectories. We're losing so many rib mussels, it really makes sense to try to stem that bleeding as soon as possible to me. Anyway, so if we look at those two species and consider oysters, there's lots of pros and cons to consider. I think on the positive side, all of these species, there's opportunities and we have effective tactics. We now know, you just saw Brian and Rachel's talk, we know now how to make baby mussels, we know how to put them back in rivers, and we'll continue to get better at it, and there's so much opportunity because they are the most imperiled species. So, most places don't, in my neck of the woods, don't even have any mussels left. A lot of opportunity, effective tactics for all three of these. There's some other interesting things. If you're looking at freshwater, if you're looking at a watershed bay program, if you could restore mussel beds up in the river, maybe you're intercepting at least some of the pollutants before they even get to the bag, right? That's a consideration. That's a plus for freshwater mussels. Rib mussels, some of my early work with Reichert-Nguyen at Horn Point and others, we determined that rib mussels, oysters can't do it, but rib mussels can filter bacteria less than 1 micron in size. And they can digest them with more than 80% efficiency. And that would include pathogenic bacteria. So we're talking about nitrogen here, and maybe TSS as TMDL things, but don't ignore that some of these animals might help with other kinds of water quality problems being pathogenic. Freshwater mussels, we do not yet know if they do the same thing, but rib mussels are super good at that. So much so that when you lose rib mussel populations near an oyster reef that's being harvested, that's bad for the oysters because they're filtering bacteria that might close down that. Right? So oyster fishermen should care about rib mussels because they might keep the shellfish sanitation, keep it open for harvest. So there's all those other considerations. Oysters, on the other hand, big plus for oysters is there's a lot of support for oysters and they are commercially valuable. On the con side, freshwater mussels, there's a lot of streams. We do not yet know if they are restorable. Mussels were lost from those systems for a reason. And because of the Clean Water Act, many of our streams, we've already tested, we've released pit tagged and caged mussels in all these streams across dozens of streams in Southeast PA, and we've had mussels hold up nicely in 90% of these. There's a few where we would not actually restore because we think that some of the reasons for the mussel declines are still there. And metals and some of the other things that are acutely toxic. Acid mine drainage, not gonna work until you deal with the acid mine drainage. So we really need a lot more work on the carrying capacity and targeting. Con, low interest, I think that'll change. Hatchery capacity, that is changing. Rib mussels, same deal, not much hatchery interest so far. There's not a seed supply for rib mussels if you wanted to use rib mussels, although we do know how to spawn them now and so forth. Whereas with oysters, these policy bans and industry conflicts are fun. So over in our system, about 10 years ago, we created a freshwater mussel recovery program, all these different things. We do outreach stuff. We just heard about the cool outreach. We're starting to do mussel gardening programs. We have volunteer survey trainings, workshops. We teach people how to survey mussels with their watershed groups, school groups. They go out, survey, and then they put data through our online database portal. But we really need and we're expanding our propagation, habitat restoration, research, and so forth. As an example for habitat, these living shorelines, again, we're working in Camden. We've been designing some living shoreline projects that include SAV beds, low freshwater tidal, low marsh, high marsh, and then mussel beds. And this site here is the Camden County Municipal Wastewater Treatment Plant. Again, these water companies and wastewater treatment companies are super, super interested in any natural infrastructure that can promote clean water off their facilities. And they're very progressive. All the roofs of their buildings are solar arrays. They're zero carbon facility. So it's some cool stuff going on. But really, we do need the propagation. So we've been working also on propagating species, and we've been working with Rachel and Brian here. Last year, we took our broodstock down here for airway floaters, and they had the herring. We did not, so we brought them down here. They gave us back, I don't know, 100,000 fresh off the fish seed and a few that were larger. And by July, they were looking like this. And by September, hold on the picture, by September, we had 4-inch animals. Born in April, 4 inches in September. And we're tagging them and using them for projects. And out of that 100,000, we have at least 30,000 still alive today that we're using for various restoration projects. So it can work really well, these partnerships from Pennsylvania to Virginia. You know, as long as we are careful with the genetics and biosecurity and all that, we're good to go. Tech is there. So this is, I think, the last slide. So we have this new program we're getting off the ground. It's called the Mussels for Clean Water Initiative for the Delaware and the Susquehanna rivers. And just, this is the hatchery and the seed and the pond rearing, like you just said, and then restoration in streams. But the other thing we're starting to tinker with, and this I think is down the road, but really love to get connected with some other research partners interested in this. Can we go beyond natural restoration? Can you go beyond restoring these mussel beds? Can you start to think about designed, engineered systems like living shorelines or something where you have sites of nutrient impairment, just like they did with the Rib Mussel Project in the Bronx, you know, but can you create aquaculture mussel farms where you put seed in, maybe 4 years later, you take the adults out on a rotating crop basis, you're actually bioextracting nutrients much more effectively than just relying on denitrification and burial. You're pulling nutrients out by pulling the animals out and maybe even the sediment from the trays, and then replacing them with more seed, and then those adults that you pull out can get relayed to other restoration sites that don't have nutrient impairments where you really just need biomass, or they could be sold for ponds, or they could be turned into poultry feed like they do in England or Norway. So these are just concepts, and I think that they'll probably raise a lot more questions and concerns maybe, but I do feel that engineering, we need to be thinking beyond. We need to really go for restoration, conservation, but we can go even an extra step beyond that. Start thinking about designs, designing natural infrastructure to achieve particular management purposes in particular places where you need that help. Question?
This particular design, you thought about utility and stormwater ponds?
Yeah, we have a proposal right now pending with State of Delaware stormwater ponds. We're thinking tax ditches. We're thinking, you know, treatment wetlands. We're thinking, yes. But we don't have any real active projects because it's chicken or an egg with funding. So yes, call us if you want to work with us. Those are all, there are so many different questions, including, right? So just to wrap up, freshwater and saltwater, this is the one thing. Freshwater mussels are no slouches, okay? No slouches. Most populations are in decline and deserve protection. We must not lose sight of the fact that we need to do conservation first. And in fact, I'll take it a step further. You know, we have these rare mussels that garner all this attention, and then we have what they call common species or secure. To me, every mussel is precious because every mussel filters 10 liters of water, you know, a day. Every mussel is precious. So where you have a bed of mussels, whether it's 1 species or 10 species, that bed of mussels should be a mussel sanctuary. Okay, there is no such thing in mussel sanctuary, but there is critical habitat designation. We're starting to go there. So right now we're looking at tidewater muppets, Lepidoptera, because the Delaware River is the largest freshwater tidal system in the United States, and so freshwater tidewater muppets, like tidal freshwater systems, we consider that a poster child for our system. And, you know, might be one of the last reproductive populations on the eastern seaboard. So any place that we can find juvenile mussels propagation, that's critical habitat potentially. You know, we need to do that conservation piece, but then also, you know, we can enhance all native species. So it's not oysters or rim mussels or freshwater mussels. They're all, the take home is they're all important. They're all important. And research and development needs, I've touched on that throughout, Joe, but you know, obviously I'm really big on this understanding the fate of what they're filtering. I think that's important. And then also carrying capacity. You know, how much really can we, what's the population biomass we might be able to achieve in different places? How patchy? And with changing climate, you know, what are we really looking at? How do we pick places and species along those trajectories? And then hatcheries, continuing to increase our efficiencies and decrease costs associated with the hatcheries. Understanding habitat suitability and how you can not just reseed, but actually improve the carrying capacity by engineering the habitat, benthic habitat, stabilizing the stormwater, living shorelines, taking out the dams, and then more, I think, thinking about this restoration of our life. The economic side. You know, if you're a water quality manager, you know, how do this is a BMP stack up against other BMPs. So, but in answer to the question that was posed at the start in the title, yes, I think investments in freshwater mussels can potentially augment existing efforts, not to replace them, not to be a magic bullet, to fix our, but another BMP in our, you know, another arrow in our quiver, yes. That's it.
Round of applause for Danielle. So maybe we've got time for one or two questions before we get our next speaker up here.
I'm sorry. Yeah.
So you talked about setting the lead versus incorporating the hatcheries. So what percentage do you think should go to prevention and living shoreline, and what percentage should go to propagation?
You know, I think that decision is something where I come up with a place and with what are you really managing for? A living shoreline provides other benefits beyond what's called enhancement. So in some areas, in my area, Hurricane Sandy, after Hurricane Sandy, all of a sudden there was a big paradigm shift. People cared about nothing all of a sudden. We've been doing living shorelines well before Sandy, Scotland monitoring, no one gave a hoot. And all of a sudden it's like, you know, everyone wants to do living shorelines. So in New Jersey, doing living shorelines will probably be a priority because of coastal resilience management goals, and we have a word called sevens and nice habits. Over here, where water quality is really probably the top driver, you know, maybe that would skew more toward propagation and receiving beds until you reach a point where you saturate your existing carrying capacity and then say, how can I increase carrying capacity? And there's policy variables. In Delaware Bay, the productive oyster reefs in the bay aren't really used to because of salinity rise. Oyster beds now are more productive in places where there weren't even oysters 100 years ago. But you can't really work on them because you can only restore oysters where they historically existed. And so I bring that up because I think that there are these impediments, policy barriers, that also factor into this decision.
One quick question.
It's not a question, but talk to me later at the service did a strategic plan on upper Tennessee river basin to evaluate various management actions. Danielle said we were interested in what's the likelihood of moving the objective or reaching our objective. So, and I think Danielle, you forgot to mention that in your mussel bed surveys, you find more, that's where you find the rare mussels.
Yeah, so we have great traction with Maryland DNR, Pennsylvania DEP, Denver from Delaware. New Jersey DEP takes this more traditional view that they don't really support restoration of freshwater mussels of common species because of concern that by pumping out a lot of Olympia or airway fluids, you might impair the remaining TME species because of competition. Right? But what our data show, at least from the tribe of Delaware, all these quantitative studies, the only place you find the rare species is living smack dab in the middle of a big bed of common species. Now that might not be true everywhere, but we think that for those species to even exist, they need to have the benefits of the stability and maybe the organic enrichment or something that is provided by a large mass of mussels or common species.
And this is also what Karen Vaughn's working and Watchtower River on she likes to say, it's good to live near the king if you live in a big river.
I think in headwater streams there are a few of the rare species. I know maybe green floaters, a few others that might be mavericks and not exactly come into play, but in the big tidal freshwater areas and big rivers, I think we can dispel that notion pretty swiftly.
Thanks again, Danielle. So last speaker today is actually not on mussels, but oysters. Julie is going to talk about some of her work with the oyster BMP expert panel, which is essentially grappled with this question. If you want to make bivalves living in the water a creditable practice, what kind of questions do you have to answer? What are the types of things you have to grapple with? And like I said, not necessarily an advocate of mussels being a BMP. I think that's an interesting conversation for exploration. But I think the questions that this panel explored and wrestled with are the same questions we would have to think about with mussels and other species. So that's why I asked Joey to come down and kind of talk about this today. I think it's going to be really interesting. Just want to acknowledge also got Jeff Cornwell and Chris Moore somewhere around here who both also participated in the panel. So some great expertise here. So take care.
Great, thanks, Joe. I was actually really excited to get this invite. I remember back in 2014, we were where you guys are at, scratching our heads. Can oysters and the various oyster practices actually be used as a BMP and get credited in the TMDL? So we sort of went down this road and we're like, well, can we make it happen? There were some obstacles. There were no shellfish BMPs that were approved by the Chesapeake Bay program. So that was the first obstacle, just to convince folks that, yes, we could use oysters as a bioextraction technology, if you will. And another obstacle is that we're dealing with the in-water BMP. So these oysters are in tidal waters. And a lot of the BMPs, when people think of them traditionally, are land-based BMPs where they prevent or stop the nitrogen phosphorus from even entering the water. When we're dealing with oyster practices, the nitrogen phosphorus is there. So now we're filtering it out after the fact. So those were some challenges in the beginning of people trying to wrap their heads around, well, can we use this biological organism? Can we use this shellfish species? And can we do this in water? So what I'm gonna do here with this presentation, I'm gonna first go over the process to have a practice, a proposed practice, be considered a BMP for the TMDL. And then I'm going to talk a little bit about some of the policy issues that we came up against with oysters and using them as a BMP. And then I'm going to talk a little bit about sort of our reduction effectiveness strategies with the oyster practices. And the good news is we do have some approved oyster aquaculture BMPs. So yes, this can be done. We can use a shellfish species in this framework. So yes, I'm gonna run through those. Before I do that, this has been definitely a collaborative team effort. We had a panel of experts that included fisheries scientists, oyster scientists, fisheries managers, conservation specialists, and we do have our panel chair here, Jeff Cornwell, which I want to thank, because when I first proposed this panel, we said, oh, we'll get it done in a year. We're almost into 3 years now evaluating oysters as BMPs, and also Chris Moore is here as well. And we have a lot of support from the Chesapeake Bay program, and we have a lot of support our organization, the Oyster Recovery Partnership, and also various folks that has volunteered their time. That's another thing I want to mention. Our panel is all volunteer, so all our panelists, they are volunteering their time to help us evaluate the science. And then we have a lot of oyster scientists who have been very generous in essentially giving us non-published data to do some of our analyses. So that has been great as well. And there's also through this process stakeholder review. So we have a lot of stakeholders that have been involved and provided some really great feedback as we were trying to figure out, well, how are we going to do this? So first, just to go over the process. So what is a BMP for Clean Water Act purposes? Best management practices, commonly referred to as BMPs, are practices or technologies that reduce pollution when implemented or installed. They can be structural, non-structural, or programmatic. They can also, as I've mentioned, a lot of them are land-based, but now we are sort of entering this sort of new world of in-water BMPs. Shellfish isn't the first in-water BMP. There were floating wetlands and also algal flowway technologies that have been used in water. And so for a pollutant reduction from BMP, how can it be credited in the Chesapeake Bay TMDL? It can be credited as either a land use change, an adjustment based on an estimate or effectiveness of a BMP, so an efficiency. That's where we focus on the oyster BMPs, is coming up with the reduction effectiveness estimates. It can be a measured reduction in direct load to land use and a measured reduction from a treatment process, such as technologies installed with a wastewater treatment plant. So I just want to kind of emphasize the word credited, because there's some confusion with that word when it's used. In the sense of the TMDL, when we're talking about credited, it refers to the model estimated amount nutrient sediment load reduction achieved by implementation of the BMP that a jurisdiction, a state, a county can count towards meeting their TMDL obligations. So it's not associated with monetary compensation. You hear a lot about nutrient trading. That comes afterwards. So when we're evaluating BMPs for reduction effectiveness, we're seeing how can we credit the reduction in the TMDL. And it's up to the implementing programs afterwards, once you have an approved BMP, to decide, well, how do I want to implement this? And they may choose a method that there could be monetary compensation, like nutrient trading credits. So kind of walk through the process. Now the Chesapeake program is a little bit unique with the Chesapeake Bay TMDL. They've decided to use a partnership model, and they're using a consensus-based approach. So for any BMPs, they have to kind of run through this process. So it starts out with a water quality assessment, and this happens nationwide through the Clean Water Act. Are we meeting water quality standards? Chesapeake Bay, we were not. We were not meeting chlorophyll A, dissolved oxygen, and also aquatic life for seagrasses and water clarity. Then a TMDL is put together, and we have a multi-jurisdictional TMDL for Chesapeake Bay. And the TMDL essentially, through this modeling exercise, will determine the amount and where the pollutant reductions are needed to eliminate the problem. And then from there, we have these watershed implementation plans where the states and the municipalities come up with plans to achieve the required fix for the problems. Now for Chesapeake Bay, the TMDL is designed to reduce nitrogen, phosphorus, and total suspended sediments. So we then have proposed BMPs that will appear on a approved BMP list that addresses how you're going to reduce nitrogen, phosphorus, and suspended sediment. So these are essentially practices and technologies that the state and local level can use. Now, with what's a little bit unique with the Chesapeake program and the Chesapeake Bay TMDL, any proposed practice, such as oyster BMPs or freshwater mussel BMP, has to go through a BMP review process through an expert panel. And essentially what the expert panel does, they evaluate the reduction effectiveness of the proposed practice following a review protocol that has been established by the Chesapeake Bay program. And once approved by the Chesapeake Bay program, these BMPs then move into a list of approved BMPs that jurisdictions can use in their watershed implementation plans. So We, right now, with the oyster BMPs, we decided to use an incremental approach, which I'm gonna talk about in some later slides, that we were like, well, there's a lot of science on some aspects of oysters and less science maybe with denitrification dealing with oysters. So we broke it down. So we actually have some approved BMPs for oyster aquaculture for the amount of nitrogen and phosphorus stored in oyster tissue of harvested live oysters. But we are still an active panel, so we are evaluating even more practices and more processes, which I'll talk about in the next few slides coming up. So the BMP review process, this is the protocol. This is simply put, there's actually, the protocol is more involved in this, but this at least gives you an idea what you're looking at. So pre-panel steps, you essentially need to have a group or the partnership interested in the practice to be a BMP. So this is a great start. Everyone talking about freshwater mussels and looking at the science, what do we know, what we don't know. That's where oysters essentially started too. And you then decide, well, is it worthwhile to do a panel to have this actually approved BMP? So the panel's prioritized, signed to a work group. We have a work group chair and coordinator. Oyster Recovery Partnership, we coordinate the Oyster BMP expert panel. And then you recruit members for your panel. And then the panel steps, once you get going, the panel will meet on a regular basis, usually monthly. And then the goal is to actually provide recommendations in a report that then can be reviewed by the Chesapeake Bay Program Partnership and stakeholders. And then once you have the report reviewed and you address any comments, then you seek approval by the Watershed Technical Work Group and also approval by the Water Quality Goal Implementation Team. Now, oysters were unique. We had several goal implementation teams that we had to seek approval. The water quality was the lead, but also the fisheries and habitat. The oysters have a lot more going for them than just reducing nitrogen and phosphorus. And then once you have that approved, then you're on that approved BMP list that I talked about. And now local partners can choose and jurisdictions can choose those practices to be credited in the TMDL. So we've heard a lot. Yes, I think everyone here, there will be no arguments that filter feeders can help improve water quality. Shellfish do filter and consume particulate matter, algae out of the water column. Now there's been a lot of work already done on blue mussels and rib mussels and other areas. And as we've heard today, so there's a lot of work also looking at freshwater mussels. For our BMP, we're only looking at one species, the eastern oyster. So that's a little bit unique from what would be essentially a freshwater mussel BMP, what I've heard. There's a lot of species. So you may take some different strategies if you decide to go this route. So we viewed shellfish as an innovative option to help improve water quality. And what helped us get started to get approval to have a panel is that there were multiple requests for oysters to be evaluated for use as BMPs to meet the Chesapeake Bay TMDL water quality goals. And it started with the Scientific Technical Advisory Committee. They had led a workshop to sort of start evaluating shellfish and nutrient reduction. This was back in 2013. Also, the City of Virginia Beach, they sent a request to the Chesapeake Bay program to consider denitrification rates for a sanctuary oyster reef BMP. That was back in 2013 as well. And there was also an industry group, the Oyster Company of Virginia, where they put forward an in-situ nutrient remediation pilot between an oyster farm and a stormwater permittee. So pretty much oysters harvested out, that's the amount of nitrogen phosphorus harvested out, and that would receive credit. So because of all those requests and the interest in the science being available, we convened a panel in September 22nd, 2015. And we were charged at first with sort of establishing a framework, how would we evaluate oysters as a BMP and evaluate their reduction effectiveness. Again, this hasn't been done. There were no shellfish BMPs on the approved list. And then once we established that framework, then we would determine the nutrient suspended sediment reduction effectiveness of the oyster practices using available science. So our panel decided that, well, there's a lot of ways in which oysters are used, a lot of oyster practices. So we should start there. We should group our oyster practices by what we would think would influence the reduction effectiveness. So we grouped them by the oyster fate, whether they were harvested from the water or they remained in the water. We looked at the fisheries management approach. So we have private oyster aquaculture where there are leases that are given out by the state to private operators who then grow oysters in a certain area. There's also the public fishery. And, go back for a second. The public fishery is, there's various practices under that, and that's more or less an area is designated as public fishing grounds, and anyone with appropriate license can go and fish. And then we have our oyster reef restoration, which are sanctuaries where there's no harvest. And there's different activities for these practices. Either you're growing hatchery produced oysters off bottom in some sort of gear like cages, or you're growing them directly on the bottom, or you're moving wild oysters from one location to another. You're adding just substrate to catch wild spat, or there's no activity. Some of these areas where you just essentially get a lease and then you harvest what's there. So the panel were looking at these practices and they said, well, it's all about reduction effectiveness. So how are we going to define that? So they pretty much defined that a practice must include an enhancement activity that could result in the overall production of new oysters, and the reduction process must occur with the practice, which I'll talk in the next slide. So in their minds then, some of these practices were already ruled out, like just transplanting one oyster from another. You don't have a reduction. It's just a transfer of the nitrogen and phosphorus. So they kind of looked at, well, there's no activity, there's no enhancement activity. So if I'm just harvesting what's there, that's the natural population. So they moved away from giving credit to natural populations and more had to be an enhancement activity that would produce new oysters and not count existing oysters. And they broke down how they were going to credit these oysters by the processes in which oysters can reduce nitrogen, phosphorus, and suspended sediment. So they looked at nitrogen phosphorus assimilation in tissue and shell, which are the blue protocols, and each of them are established at their own protocol that we're going to define the nitrogen phosphorus reduction. And then enhanced denitrification associated with oysters, that's unique in the sense it's pulling all that organic particulate out of the water column where it could cause problems, become bioavailable again, and is biodeposited to the sediments, to the bacteria that can process that, and then hopefully the goal is lead to denitrification and the release of the nitrogen gas, so that it's another removal process. Those are the ones we've been mostly focusing on right now. There are some other protocols associated with burial, and then we do have sediment reduction associated with oysters. There's not a lot of data for that, so that's sort of on the bottom of our priority list to evaluate. So the ones highlighted in blue and purple are what I'm going to talk about in this presentation. And then back to our framework. Again, there was a lot of concern about using oysters as a BMP, and a lot of it stemmed from, well, there's a lot of variability. How are you going to come up with an estimate that addresses the variability of oyster growth, survival in these locations, and ensure that you're going to meet the water quality objectives? So the panel decided to create some steps. Their first step was what I talked about, is pretty much separating this out, identify practice oyster associated reduction processes suitable for BMP consideration. Again, focusing on enhancement activities. Then they looked at whether, well, is there sufficient science that exists to actually determine the reduction? Can the quality and scope of data, is it reasonably constrained, where we could have an estimate? We leaned more towards the conservative side since we were dealing with a biological organism. We want to make sure that data could support that. And then is the estimate verifiable? So that's very important through this process. With any BMP that you're putting forward, you want to make sure it can actually be tracked and verified. And then any identified unintended consequences, we asked ourselves, well, are they manageable? Can negative effects be addressed so they don't outweigh environmental benefits? So this is sort of the framework that we were operating under. And this led to 96 oyster practice protocol combinations that the panel is actually evaluating. So I do recommend for freshwater mussels, after hearing that you're dealing with over 50 species, because initially I was thinking, oh, maybe you would have all species. No. We dealt with 12 practices, and it sounds like maybe from a practice standpoint, you're maybe looking at maybe 1 or 2 that you're evaluating. That's reasonably to address. But the goal would be to see if you could find a way with all the different species, and as people are mentioning, looking at a mussel bed and not having to have reduction effectiveness based on species, because that could be quite timely. So if the data supports actually where you could have a BMP where you can lump different species together, that would be definitely the way to go. Because even with 96 combinations, that was quite a bit to tackle for our panel, and that's why we're going into 3 years instead of 1 year. So I'm going to be talking about some of these, but before that, I just want to sort of bring up some questions that have been coming up using oysters as a tidal and water BMPs. So our panel is really, what we're charged with is really looking at the science-related questions. So does existing science support nitrogen phosphorus reduction estimates for the various oyster practices occurring in Chesapeake Bay? And how can this reduction be quantified and verified? Again, that's important for this process, given the variability in oyster growth and survival. However, through this, we have come up with some policy-related issues, and most of them have centered around removal versus sequestration and the type of activity. So when we're dealing with the in-water BMP, and especially for the oyster restoration, where oysters aren't being removed, they're staying put, we asked ourselves, well, can the buried biodeposits and sequestered nitrogen and phosphorus in the oysters receive credit concerning oysters that remain in the bay? And we also asked ourselves, and this may be unique to oysters, this may not be an issue that you have to address with the freshwater mussels, but with oysters, our stakeholders brought up the concern that, well, if you give credit to the amount of nitrogen phosphorus stored in that shell from a harvested oyster, the goal is for that shell to get returned to the bay. We actually use shell as substrate for these oyster practices. So did you just credit something that just got returned to the bay and now you really didn't remove any nitrogen and phosphorus from the system? So that question came up. Can you even give credit for temporary removal? And that's something we're still trying to address. And then a new one that has come up, can conservation practices with no physical activity be considered a BMP? So this is where you designate an area as a no harvest zone. And you're pretty much doing that to preserve existing oysters, and you're not planting any more oysters or substrate. So the activity of actually designating an area and telling folks, hey, don't go here, don't fish here, is that enough to qualify as a BMP? And that's something the Chesapeake program has been looking at now for other practice, like land conservation practices as well. So we may be seeing this evolve more over time. Do we have a quick question? Yeah. Right.
Programs within the service that give some sort of benefit to the landowner for, say, assigning land and not.
Right, and not touching it. Mm-hmm. Yeah, so we may see in-water BMPs, maybe something along those lines could happen as well. Discussions are still ongoing with that particular issue. So sort of our status, one of our stakeholders actually brought up with the legality of in-water BMPs from a nutrient sequestration standpoint. And if it remains in the water, is that even legal through the Clean Water Act to give credit to that? And we worked with EPA lawyers for over a year, and we actually have a legal opinion from them now saying, yes, in situ BMPs are legal since pollutants are removed from the water column. So where they're causing the problem, they're being removed. And this assumes the practice involves native species. And then how to count the in-water BMPs towards the load allocation, that came up as well. And this has been resolved. They said, for now, it's not credited to any specific source sector like wastewater, ag, stormwater. Instead, it's going to go towards the total non-point source load allocation. And again, with the conservation practices, we're working on trying to resolve that one. So like I said, we have actually approved oyster BMPs. There are several oyster aquaculture BMPs approved by the Chesapeake program. We have a report on this. And this is for the amount of nitric phosphorus stored in the tissue of the harvested oyster, and they have to be alive. And the endorsed practices, off bottom culture, and bottom with substrate addition. So we have some potential oyster BMPs that may be approved in the fall for our second report. We're looking at nitrogen phosphorus assimilation in tissue of harvested oysters from public fishery practices that use hatchery-produced oysters. We're looking also at nitrogen phosphorus assimilation, so sequestration in the tissue and shell for oyster reef practices, now that we have that legal opinion that it is legal to look at that. And then nitrogen reduction via enhanced denitrification associated with oysters for oyster reef restoration practices. And if you have time, if you're interested in denitrification, I point to Jeff Cornwell. He's our expert on that. He's been helping us really work through how can we credit this in the TMDL. So I'm not going to go through all of these, but just give you kind of a sense. These are the key components of the BMP report. You need definitions for your practices. You need how you're going to establish a reduction effectiveness. I'm going to go over, if I have time, maybe a couple of strategies. I have maybe 5 more minutes. Yep, okay. And then land uses, where it applies. So for in-water BMPs, where the nearest shoreline segments. Load source addresses, we're non-point source. Baseline description, so baseline is really important. The goal is not to give credit what's already there, is to give credit to the activity, so what you're putting there. So something to keep in mind. And then qualifying conditions, when would this practice qualify? Useful life of the BMP, I was like, wow, freshwater mussels can live to 100 years. May have a very useful life in that category. And application and verification guidelines. It has to be, you have to be able to verify and report it. So we've looking at, let's go briefly, some strategies to determine the reduction effectiveness. For harvested oysters, luckily we have some solid relationships between shell height to tissue dry weight. And we have a lot of data on the percent nitrogen phosphorus content in tissue. So we pretty much said, well, we can use these relationships to convert shell heights, which is easier to measure from a verification standpoint, and then be able to convert it to dried tissue weight. And then we can use an average percent content to figure out how much percent nitrogen phosphorus is stored in the tissue. And we're using that same strategy for shell, and we're using that same strategy for verification. For oyster reef restoration, where they're not removed, we're using those relationships, but the only difference is that for like a harvest oyster, every harvested oyster I can get credit for. It's a continuous credit. But for oyster reef restoration, those oysters, we can't double count. That's one thing you can't do in this process, is can't double count the credit. So we're essentially setting up where it's going to be based on biomass. And so we're trying to reach a standing stock that the reef, we're trying to restore it. Say if our goal is 50 grams per meter square of oyster dry tissue, then you get credit for that. And once you reach that, that's it. You know, you can't keep getting credit for that same amount of stored nitrogen and phosphorus. So we have some strategies involving that. And then denitrification, where the panel did determine we need site-specific rates under known oyster tissue biomass. The denitrification data was so variable, there was no way we could establish a default estimate. So we're saying essentially we're going to propose a method in which you can actually come up with your estimate, but we can use a default approach from a biomass standpoint to verify it. So there are strong relationships between the denitrification rates and biomass for tissue. So this is kind of an example of what that looks like. We're using regression equations. I don't go in that too much. And here are our approved BMPs. It's based on size class and the type of oyster, whether they're diploid or triploid. Diploid oysters have less tissue than triploid oysters. As you can see, these aren't astronomically high numbers. So I would like to reiterate the point that Danielle mentioned, is that we're not seeing these BMPs as replacing land-based BMPs. Essentially, we're seeing them as another tool in the toolbox that can maybe help from a local standpoint. So this is actually in draft. We're working on this report, and you're actually the first group seeing it. So this is a sort of a sneak peek. This is dealing with oyster restoration and the amount of nitrogen and phosphorus stored in the reef. And we decided we wanted to do a partial crediting approach. Again, so we're trying to meet a standing stock, and we're only going to get credit for that. So for instance, if the goal is to reach 50 grams per meter square of oyster dry tissue weight, we have our enhancement activity where we plant oysters. Our baseline's at 10, you get 0 credit. Then I measured again at 20. I would only get credit on the appreciated biomass, which is 10. But as a buffer, because we don't know what's going to happen to that reef, if it's going to decline at all, we only get half of that credit at that time. So you only get credit for 5 grams of the biomass. Then the next year I look and verify my biomass, I'm up at 50. I'm at my target. But I still don't get full credit yet. I get the remaining here I had here. Appreciation was 30, so I get half of that. So 15 plus 5 gives me 20. I only get credit for the 20. And then, oh, something happened in year 3. I've lost biomass, so I get 0 credit. And then my following year, I did another enhancement activity. I planted. I'm now back to my 50. I get my remaining credit. So overall, minus the baseline of 10, I should only get credit for 40 grams of oyster tissue, how much nitrogen and phosphorus is stored in that. So that's sort of the strategy the panel is taking, to be conservative, to allow a buffer, and if our reef all of a sudden declines in its populations, and to not overcredit. That's what we're trying to avoid. We don't want to overcredit. And same with enhanced denitrification. Let me skip that slide. Same kind of approach. We need a site-specific estimate. So we have measured denitrification rates. Enhanced rate would be an oyster site minus your baseline rate. Do this at different time periods, at least 3 replicates, could be more depending on the size of your operation. Dark incubation is needed, and light may be needed. If light hits the bottom, that can influence your rates. And again, looking at oyster biomass, because there's a relationship between denitrifications and biomass. And again, different sort of crediting scheme. You get full credit if you're at the biomass. Your observed biomass equals the biomass you establish your rates. If your biomass increases, we don't know what the rates would look like. So to be conservative, you would still just have the same level. So your nitrogen credit would remain the same each time, unless you created a new estimate at the higher biomass. If it declines, it can decline linearly until at one point, if it's less than 50% of the biomass you observe, you get no credit. So we're trying to establish rules that buffer against overcrediting, given the variability in oyster growth and survivorship. I'm not going to go over that. That's shell, so I don't think that's as relevant for this group. And last thing I kind of want to point out, it's important in this process, we felt to offer flexibility to the implementing programs. So for our first set of recommendations in our first report, we gave different options that you can use those default estimates, but say they didn't want to do the verification measures to see what size their oysters were. Well, there's regulations in place. You can only harvest a 3-inch oyster. So you would only get credit for the 3-inch oyster, even if you had 4-inch oysters, if you're not going to do the verification. So that's the most simple approach. Option B, use your default estimates, but do the verification, measure your oysters, and you may be able to get more credit essentially by doing that. Option C, we have, in all our BMPs, we always offer for the defaults that you can establish a site-specific estimate as well, because our default estimates are very conservative. So your particular site, you may have oysters with more tissue than what we're showing in our default estimates. All right, so for conclusions, we feel like having a framework that kind of breaks it into parts is a smart way to go, because we actually got approval for some BMPs as our panel is still working on it. The endorsed private oyster aquaculture BMPs are actually our first shellfish BMPs approved for use in cleaning up the Chesapeake Bay. Jurisdictions can select them. Again, they complement, not replace land-based BMPs. This opens the door actually to oyster aquaculture industry to participate in water quality programs, as you guys were mentioning, like engagement activities for freshwater mussels. it is great when you have this on this approved list, because then, you know, it's another tool in the toolbox to even engage with folks and get them interested in working on these water quality programs. And it could also provide additional business incentives. Oysters are a commercial species, so there may be some monetary compensation involved with that. And again, any policy issues and implementation procedures would need to be addressed and developed related to the application of these BMPs. The panels are just responsible for determining the reduction effectiveness. And I just like to stress, you know, if you do choose to do a BMP panel, flexibility can really help, especially with implementing programs, being able to go forward in choosing your BMP to use. And I'll stop there.
Thank you, Julie.
Yeah, thank you.
One or two questions? Okay, so yeah, it would be great to have a freshwater mussel. This is another question, I guess. So how do folks feel? We have enough information to start acting now, or do you think there's still more to do technically and scientifically?
That's a good question.
So
for
for open question, hold on just a second on that. We'll try to merge into that. I know we're kind of running on overtime here. We've got a crunch session because we are still planning to go to the hatchery for those interested in taking a tour. But we can kind of get to that. Any other just kind of clarification questions on the presentation?
Yeah. I'm going to make it a clarification for you guys. So how can you say you're only looking at one species if you're looking at in situ models of oyster acres that you have up to 300 participating species in that recommendation?
Right.
How do you treat it only based on oyster length of the sample of that reef?
So the denitrification estimate would be a little bit different because there are two different main approaches that you can do. You can look at sediment cores, which you're not looking at the whole community like that. But Jeff, his lab, they do whole community observations. So that essentially, that estimate would combine those other species. And we were focused on just oysters, but I think from a denitrification standpoint, and Jeff, correct me if I'm wrong, we're okay with counting the whole reef.
And that's exactly what we do. We know all those other species, there's mussels, lots of other filtering organisms are attached to those reefs. And for the most part, we can hang our hat on biomass to predict denitrification, except it's a lot better when you start considering those other organisms, including polyps.
So that goes back and, you know, not doing species by species. We started out looking at oysters, but from a reef standpoint, it's better just to give credit to that whole reef. Those measurements are quite expensive to measure denitrification rates. So it's better to get that measurement and use that as your BMP. Again, all that, the draft stuff, we have not put forward for approval yet. So we could hear different things from the Chesapeake Bay program, whether or not we can do that or not. So those are in draft.
Well, thanks, Julie, and thanks again for all of our speakers today. Maybe another round of applause just for everyone. Really great stuff. So I'm trying to just designate, 10 or 15 minutes just to kind of, hear back from this group. And there's a lot of diverse expertise, and I just want to kind of bounce these ideas off you and hear what you're feeling right now. What are you thinking? What are you excited about? What are you concerned about? And what might be a next step? What's the next step that makes sense or one that might not? And for folks on the phone, so you can type this into the chat window and we can feed that back later, and we'll synthesize all this and send it out so people can see kind of what were the opinions expressed here today. So we'll just start with 5 minutes on the concerns. So what are the concerns that folks have on moving forward with dovetailing mussel restoration and Chesapeake Bay restoration work?
Well, one would be the political climate in terms of mitigation, regarding the water protection and wildlife.
Okay. Yeah, Jamie.
I have a question that you like out of there's some barriers for NGOs for species. This is the area that covers those populations. How do we overcome that?
apply for a scientific collective permit.
Not section 6.
Section 7, scientific collective.
Other concerns? Oh,
I'm not sure I can articulate this very well, but when I think about mussel restoration and sort of feasibility and opportunity aspects. So if you think about mussels versus fish, it's a lot easier to produce 1,000 or a million herring and yet, most of our fisheries restoration efforts were not successful. And so it's easy to produce a lot of fish, and for things like Hatchery and other things, we weren't able to ultimately restore them. So then what is the hope that we can do that on the mussels side? It's how much more difficult it is to produce mussels.
so concerns about success long term of the effort. Will it work?
Yeah, so
Given some other background.
On the early side, we see significant numbers of individuals. And then I guess part of that too is the opportunity. So a lot of sites that I work at seem to have no mussels at all. And these are urban sites. So is there actually an opportunity? If they don't have mussels, because say 10 or 20 years ago, water quality was so bad they got wiped out. So then clearly there is an opportunity. But if it's because the water quality is still so good that they can't survive there, we don't actually have the opportunity. So I like to spend on second.
Okay, great. Greg.
On that, that's what that's there's a day other issue facing we can produce fossils now and get it in the watershed. We don't know, there's so much we don't know about habitat, combination with water quality issues, food needs. What is it that's keeping the multi-stressor combination, bacteria across a lot of different things? And what would be, there's so many other aspects of this, but what would be the biggest impact of that besides one thing that we still need with the riparian buffer protection would be water quality understanding, better understanding. Would be a mobile lab in the field and pump the water in that stream through the lab while you're studying. There's so many different aspects.
Right.
That's part of the data.
Yeah, right. Danielle?
I just want to quickly respond. We've been focused on that question for 10 years. And in fact, even when Matt was my master's student, he mentioned that earlier, we were doing caging to gauge stream readiness for restoration. All those questions like, can you go that next step? Should you? And I'm not saying we have all the answers, but we have perfectly fine happy mussels 7-8 years in some of our streams where those questions come up and they're doing great. But I would answer that the biggest problem we still have, at least in my neck of the woods, is stormwater and benthic stability. So it's all about, decreasing the benthic shear velocity. Most of the water quality and food conditions will support mussels. It's mostly physical bed habitat constraint. And that is fixable by dealing with stormwater.
We also have some ion balance issues to study the stormwater, not always.
John?
Yeah, I think it was Brian earlier that talked about how the majority of the funds that are available for restoration efforts are mitigation funds, and just thinking about that in terms of that, I know a lot of the mitigation funds come from Dominion, and we continue to deal with the problems that are making those mitigation funds available. And so, then, what is like that cycle, how do we get out of that cycle? And,
So there's a lack of funding right now.
It's a lack of funding that we're relying on as mitigation.
Right, it's damage
The
recovery.
damage continues,
Yeah.
and so.
Maybe two more.
So, big kind of policy concern with any in-water EMP, including the oyster one that's still under development, is Not all BMPs are equal from a space and time standpoint. Just like you heard, there's no stormwater BMPs to stabilize the shear. You can't use freshwater mussels as a surrogate. And you also, if you're cleaning up the water after it's become dirty, may be fine from the total Bay TMDL standpoint, but does not satisfy local water quality TMDLs for those discussed. So I have a general policy concern that just putting another arrow in the quiver without some policy guidance as to which arrow should go when, across the entire Chesapeake program, or anything else is trying to use this. As we're getting into considering institute BMPs, we should probably take some time.
Okay. Jeff, and then, so I'll just mention, if anyone wants to email these to me later, happy to pull that in together. And no wrong answers here today. This is just, like, what's your reaction? Because if you're feeling that, someone else probably is too, and, either it's a valid concern or it's something that needs to be answered. So one way or the other, we need to know about what these reactions are. So Jeff.
I think that the biggest piece, so Danielle did a great job of saying this is how much material is brought this filtration. The thing that materials would be where your water quality gravity and understanding that assimilation, burial, purification, maybe it's going to get remineralized and show up mineral algae or something else downstream. I think that balance is sort of the next frontier if you're going to figure out whether there's that removable nitrogen. Having said that, when we move to the sites,
great transition. We're ready to move over. Put on your optimistic hats.
So the application that we look at sort of a stream environment versus the bay. In the stream, you got that nitrate coming from the watershed. And the limit to denitrification at the bottom of the stream usually is organic matter deposition that pulls the oxygen out. And so the opportunity in freshwater, if you can amp up in the mussel bed, the respiration rates in the sediments, you can denitrify from that nitrate in the water column. Move down to the bay where oysters are, you convert that to particulates, basically algae, and you filter that, and the limit there is something totally different. That is the nitrate. You can't get the nitrate because there's so much of it that you don't have the oxygen in the sediments. And so both places should, we know oysters do a great job of denitrification. There's no reason, even though the mechanisms that freshwater do a very good job of this, but different mechanisms to take the nitrate out of the water.
See how that is? No, but that's a great point. Yeah, so other things that excite you about the world of mussels.
Again, the urban settings, it's interesting, there's not a lot of opposition because in the short run, it really looks like all those issues don't like that. I mean, trying to sell the wire improvements, start talking about transport plan, storage, that doesn't really sell them well. Absolutely short. But if you say, well, the business is part of this world, I would think that would actually be very convincing. And so, if there was an opportunity to sort of bridge those two things together, stream restoration is almost always done with geomorphology, solely with geomorphology. But now you're putting the big that is biological, that's possible. That would be one of the things that you have is just next steps would be maybe to bring in somebody like Mussel to provide a solution and ask them if we were to do a mussel restoration to see what you would support. Are there some special accommodations? These mussels, or is it just what you're already doing, but also work?
So other things you're excited about, Craig?
I'm more familiar with the I would do that on the stream, but warehouses and outside. And the capacity over the last 10 years has grown so much through basically safe streams. And I think that we will follow that with numbering them, studying them year in and year out. And I see today that those things, I think that's exciting.
Yeah, I want to, the whole thing excites me. It's exciting time to figure out freshwater mussels to the bay, oyster restoration. And even when I got started in my master's thesis, the only place I know was the marine literature to learn how to do anything, how to come on my feet. So, but I realized a couple of things. Being an hatchery girl, actually, but the service and state partners in the Northeast, Maryland River, Massachusetts has witnessed the highest returns of kill life in decades. And that's because of this hatchery program, truck and transport, so they're collecting them as they come up the dams. They hit the first dam, there's a big fish rate, the first dam on the Merrimack River. We've had an extensive truck and transport program, and up and over, stocking them out in the tributaries, and because of the in 10 years of doing that, in 5 years seeing they have the highest run this year is at 400,000. They just stopped counting because the volunteers were like, we committed to this over time.
Right.
So when I say it does work, hatchery program do work, successful. They kept on right now within the states. It just takes time. You have to commit for the long term. Because these are long-term rooters where they delay reproduction. So it is possible. I just want to add quickly that there are some species that are much hardier, that grow much faster. It's a different game of fish. For some species, we're there. We're very capable of different sites, but it's
yeah, and I think it's large that although there's much greater effort for the individual, you are increasing them at a stage where the features of ours is much more level. Compared to the parent program, like they were stopping so and never thought that they were all right, and it's here they were. And so obviously there's much.
We're also doing this massive program with families, both fish and
So maybe anything else that excites people about working on mussels?
So the potential to propagate edible species is always the big for the food writer side of me, to the degree we can replicate the first program. I know that's once you start talking about mussels.
Yeah, no, I'm actually surprised that question didn't come up. Your people eat them. Any other last exciting things, Chris?
Just real quick, critters excite me, and most of us in this room are scientists, so we're excited about this anyways. But the general public is not always excited about egg restoration. We talk about low to stream with rates and things like that, but you throw a critter in front of them, whether it moves or not, you get excited. So this makes you excited.
Oh, they move. They got those lures that look like it's a magic display. Okay, one more. Any other exciting thing?
I'm excited about the opportunities with COVID. We've largely talked about water quality benefits. There are all these other benefits that go along with it. Increases the habitat, increased biodiversity. It's an opportunity to do restoration in a lot of locations for some species.
Okay, and one more.
I'll just add on to what Matt said, but the connection between living shorelines and the tidal, the mid-tidal species, and the connection between riparian buffers and freshwater species, those are B&Ps that we get behind and we support. We know how to get funding for. There's a way to co-locate those VPs because they are natural memory, that's going to make our message stronger compared to some things we have in
Yeah, so great transition, moving into next steps. And, no bad ideas here. Like, this is like any potential next steps we could consider. So, Danielle.
That's exciting.
Yeah.
You've heard a lot of my excitement on science and restoration stuff, but I did want to say it's exciting to me. It's sort of dawning on me now that we have all these juvenile mussels, that there is tremendous opportunity for outreach. We're looking at mussel gardening. I mentioned that earlier. Engaging these schoolers. The point was just brought up about PMPs, where you can get kids to help plant trees or area, right? And you can give them little hatchwork polytested. Give them some baby mussels, just like oyster gardening on telescopes works really well to engage private landowners. I think it's exciting to me to think of all the ways that we can use this
I'll add one more, actually. I'm sorry. So one of the things I heard from Rachel and Brian was that the last 15 years has brought a transitional period of success. That where we are today is not where we were 15 years ago. And, you never know when you're going to hit that curve and things really start to accelerate. But, it seems to me the time is ripe now, at least that we're in a lot better situation today than we were 15, 20 years ago. So, be probing whether it's possibilities.
Bill, can I just add a benefit?
Yeah.
You have to jump on it. Just got to make sure that it's really and it's showing how they want to find instance around that muscle stuff. For the drivers, and they know that it is muscle beds provide good habitat for fish that we don't want to go fishing for. So your communications, doing outreach, that's another the book I use.
So great. So next steps, like what are the things that might come about? Julie, then.
Yeah, what we did, Oyster, we have before we came in on the program to get panel crew. We did, like I said, the literature review, put it all on play. Say, well, what literature, what data do we have? Can we actually do this? So it sounds like from the white paper that we developed, kind of fits that. And then so really, I would say next steps, people, the white paper fill that, or do we need to supplement it with more maybe research? And we can look at plan projects or ongoing projects, not just published research too. Where we reached out to folks and talked to folks and said, can we actually say how much they can cost for this work issue? Can we say something about transportation rates, hydrogen, food? So we tried to find all that information, put it on one spot, and then we went to our Fisheries program and said, hey, we feel we do have enough data to do this, and they were agreeing. So have it all on one spot.
Yeah.
I was going to say for data next week, this information, you guys communication center now, or so much, we can easily measure that. So I'll just try that.
Yeah, exactly.
So why? Because I've written proposals to do that 5 to 10 years and never gotten them funded. So it's easy to say less easy to get funding for it. But yes, I'm writing that. But picking up on what Julie said, I just want to say we have this white paper coming out. It's really focused on the gross. It's not really a review of, because we have a separation division to take up here, but and registration rates and all that. But we don't have denitrification rates. We don't have nitrogen concentration shells. There are research needs that need to be funded. What I would propose on the next step would be a second workshop like this, where the scientists primarily get together with the BMP people like Julie and to clarify what we do with development. Get the white papers done, but then maybe a second white paper done. I think we need to clarify if we're worried about the BMP, what specific research needs that still need to be filled. Set a research agenda and then find some way to find funding for that. If we really want to move this forward, because I've been ranked at NSF and and NOAA and EPA, and I've just never been able to move the needle on this. And I can't go to the freshwater mussel world because they're not yet sold. You know, the freshwater mussel conservation community is not yet sold. So where does the funding come to address those research needs for PMC setup? I don't know.
No, look, if we break down the property that we did in the Delaware protocol, we found we had a lot more data with discrimination than the expectation. So you can essentially move forward with the animal. So he's not covered out part. And through the panel process to identify research gaps. And we've actually, as a panel, there's researchers on our panel who are doing research projects already to identify research gaps and to address research gaps identified in our first report. So that may come out at the end of the summer. So sort of this ongoing process to support, like, is we have to out where we have data, and when we don't identify, and then we can maybe fill the gaps as we go.
So continued research is needed. That's one kind of sensitive thing I can say. And let me just add, so and take this a step up in terms of like a research need. What do folks think about the idea of asking the Chesapeake Executive Council, which is states from all over the watershed, to include a directive that's not specific, but that just says something like what the Obama executive order says, that, you know, explore pathways to help restore mussels in the Chesapeake Bay watershed. That's an end goal that we want to see happen through our Chesapeake Bay watershed efforts. Do people have any problems with that?
I prefer that over trying to shoot on into all our quality.
Well, I mean, honestly, I don't think that they're necessarily mutually exclusive.
We don't even acknowledge that we go right to helping conservation.
But so my thought, how does that connect to research? You know, then people researching can say, well, why are you doing this? Well, there's a directive from this very wide, influential body that says that we want to do this. You know, it gives researchers a carrot or a, whatever it is to hang out there, a deliverable for their various funding sources.
Yeah, one thing that I always ask is why, right, the plan, like the project or the funding source, whatever, justify why you're doing something.
Any other next steps, Paul?
In the short run, workshops. You know, so one source of funding would be workshop. The research is pretty high success rate. Right, you have someone such as yourself who really not forward and but that would be a way to fund for the future leaders.
It's a great idea. Sure. It's a great idea.
Yeah, I noticed on Julie's slide, it started with
It started with that workshop, yeah.
each one of her.
Yeah.
So, who sets the agenda?
That's for my time, we're gonna have to do, but, we have to mark the time, I'll do that.
Jamie, next step.
Funding is more guidance to get into sustainable funding mechanisms. So a goal, starting point, we can get a goal for what is needed and get out of the reactive restoration, mitigation, or correction practices coming out of our areas. So I think getting people together to talk about what is the real what we start with, building more conversations with decision makers like my pipeline.
Great. So we're about in overtime, and one more next step.
It'd be really helpful for NGOs to know which streams are suitable habitat now, like you fish.
Identify where we can work.
So one thing that we do with a lot of watershed associations is we give them hit tag lessons and practically engage them in help do tag. And then we go out with them to pick suitable habitat in the streams that they're interested in, and then they help us monitor these populations. It takes time, but over years we've been able to learn a lot from that. And what has been astounding to me is some of the urban streams, like Tacony Creek and Philly, which is rarely going out, mussels did really well. And in some streams, where they did pretty well, they didn't persist mostly because it got blown out by the flood and storm. But something to consider. These source mussels, kind of the seed that can be produced at these hatcheries. Just, there's, I don't know if you guys do, that kind of stuff, Rachel and Brian, but it's a nice way to engage people and get them healthy.
I think it'd be good to think about which of the Bay program gifts or workers might be interested in this stuff. The Bay program.
Yeah.
Yeah.
So we're already a little bit late on going over the tour of the hatchery, and I don't want to cut that any short. So I know a lot of people travel a long way to get here. If you have more thoughts on any of these categories or anything in general, feel free to send it to me, and we will try to synthesize this into something that's valuable that people can look at and comment on. But thanks to you all for coming out today. And I know this isn't really something that's probably on a lot of people's work plans. It's sort of just spontaneous conversation. But I've learned a lot today, and I'm really appreciative of everyone's participation. But before we go, I just want to acknowledge Liz has really helped, make sure we all get fed today and logistics, and Jack Ryan as well, our intern, just pulling this all together.